session, which is, I think it's on health and medicine, thanks. So I want to talk about the health and medicine domain, which is our newest domain. It hasn't even had its first birthday yet. And I guess when we have a look through, you'll see the start dates for many of these are very recent indeed, but I think you'll see there's also fabulous progress in a number of those sorts of things. I'd really like to thank all of the people too who contributed to this talk and who will contribute to this session as well. So I think everyone's very much aware that when it comes to health and particularly around infectious disease, that there are a number of factors that are contributing to the current outbreaks that we see and the spread of disease. And what's really necessary are, I guess, new approaches to be able to deal with these types of things. And it really comes about because of, I guess, that need for us to think differently. And SynBio approaches really give us a great opportunity to take a different approach to be able to deal with these sorts of threats. And really what we're talking about here is the provision of the maintenance of good health along with prevention strategies, treatments that are there, and these types of things are really going to help alleviate not just the burden of illness but also economic burdens that currently exist. As I mentioned just before, synthetic biology has a significant role to play in future approaches to both human and animal health. And really it's about that. It's about taking a different approach to be able to provide new treatments. And as you'll see as we speak through this, focus on how can we deliver better approaches to vaccine manufacture, production, new diagnostic strategies that can be put in place, and the development of new therapeutics. And again, it's really about, as it says at the bottom there, exploiting our synthetic biology capability for the development of novel strategies. So within our health and medicine domain, our goal is to deliver the tools to be able to detect, control, and eradicate diseases that are of high importance to us. And that really encompasses our ability to build capability and capacity to advance SynBio in this area. It's about developing new innovative strategies and platforms, and obviously we want to develop those in a responsible fashion so that they will be adopted in the future. And it's about taking an approach that can transform the very nature of these. So how are we planning to achieve this? Well, it's about focusing those synthetic biology capabilities and intensifying the research that we have on projects that face around the host pathogen interaction and how can we modify those approaches, particularly in the areas of biomanufacturing and product development for vaccines. Again, about understanding that host pathogen interaction and interface for the development of new therapeutics, and again, the production of those types of new therapeutics. And also, what can we use and what knowledge can we develop from this to develop approaches that can help us to harness aspects like the host response and therefore associated biomarkers of aspects of that response to develop new diagnostics, and particularly to enhance existing diagnostic approaches. So the project areas that we look at are really in the areas of developing those targeted approaches. And as I said, it's about trying to advance the strategies that may well exist already, but also developing new platform approaches to do this. And it's about specific targets and discovery and control of pathogens, or in the various reporting systems that may well be used in these diagnostic approaches. But also, and we're all well aware of this, I think also, that the changing landscape that we have with regards to how we treat infections, particularly the use of antibiotics and antimicrobials, there is a current emphasis on new strategies that can reduce the reliance that we have on these. So clearly our platform has the potential to add to the set of tools that we have in this area. So it's about the uptake of advanced technologies. So what are the facets that we can use from these sorts of things? So it's about enhancing protective responses against a pathogen. It includes aspects such as, can we interfere with host and pathogen in some way? Can we use the relationship that exists between these and modify that relationship to block the infectability, or even to reduce the type of response that's there from what may be a deleterious response, but to modify that outcome so it becomes towards a more beneficial outcome for the host and that. And these types of strategies and it's not just a one-way street, and I guess all of the projects we have within this domain talk to each other in many ways. So there's just in the same way that there's a host pathogen interaction, the research that goes on within these also has a host pathogen interaction in there. And it's about, can we develop new aspects that can synergize together to help to reduce, shorten levels of treatment or lower the needs that we have and improve the outcomes. So within this domain, there are, as I said, it's a new domain, and there are a number of fellowship areas, 3 projects, and a number of PhD top-ups that are there. And they are broken up into 2 clusters that we look at, and as you've seen from what I've described, those are clustered into the approaches that are directed towards the host, so host-targeted diagnostics and therapeutics, but also associated with that is the activities which are directed towards discovery and control aspects. So for that first cluster of projects, it really is about, as I've said a couple of times now, about focused approaches to diagnostics and therapeutics and ameliorating the outcomes of disease. The extended future we hope from these is to be able to develop processes for the development of small molecules, or even for assistance in the repurposing of existing drugs, development of biologic strategies, and as I said before, about enhancing vaccine production strategies, and really directed approaches that work towards aspects of not just curing disease, but also, as I said before, about ameliorating the impacts of the disease that goes. So our host targeted aspects really involve 7 projects that are there. Now, a number of these, or certainly here, the platform Z is presented as a poster on the other side there, so I certainly recommend that you have a look at that poster or any of the posters that are here. And we'll also be hearing several talks subsequent to this one, which will fully describe these. But I'll just highlight some of the aspects of the other projects that are not being shown in that way. So the first one I'd just like to talk briefly about here is Tara's project here within this, which is again about synthetic biology approaches to engineering mitochondrial genomes and about improving production and therapies. This project is just recently underway, but it uses synthetic proteins as tools to edit and engineer. And it looks for a minimal transcriptome within this. Again, as I said, it's recently started, and they're making some good headway in this area, and it's about delivery of genes for the mitochondria. Oops, I touched the wrong thing. There we go. Another great, and I think really distinctly different project that we have within this is the Ticker project, which is about a cellular recorder. So its objectives really are to be able to use synthetic biology technologies to be able to monitor individual cells and the process of interactions and activities that the cell has gone through within this. It's certainly a unique and distinctly different approach to many of the medical activities that you see. Again, it's another project that's just recently started, and Tom's here, and so I would certainly encourage you to have a chat to Tom about this project. But you know, it's distinctly different because it really looks at the individual cell rather than in our normal approaches that we take to medicine. We look, you know, very broadly about the response, but as we know, it can take minor populations of cells that are responding that can actually drive a particular response, whether that be beneficial or deleterious. So this type of activity may afford us you know, the ability to look very specifically and very targeted, in a very targeted fashion, at the type of response that's going on, and therefore tailor-make appropriate responses to direct those cells. An excellent project, I think. Next is what's known as Platform Z, which is Z chromosome edits and the D for again. Edits and...
It's been so long now.
Something else. I can't remember. Anyway, this is a project where you think differently about the egg. So people are probably aware that for the manufacture of, say, influenza vaccines for humans, we use the egg as the laboratory as the biofactory for producing vaccines. But essentially what's used by industry is an egg that was designed just for food. And it just so happens that the medical industry also uses that same egg. This is about thinking differently and saying, can we design an egg that's actually built fit for purpose, fit for the purpose of being used as a biofactory, rather than just using one that was being designed for food. And this project's moved quite dramatically, and it really uses that concept of taking a biobrick, so a gene, and inserting that into you know, the avian genome in this regard, and modifying that environment, that biofactory within the egg, to produce a vaccine that's more appropriate and more effective for use. You may have heard it that recently there's been a lot of controversy around flu vaccines not being specifically designed for human use because they adapt to the egg. This project uses you know, a gene to modify the expression of an antigen expressed in the egg, and allows that vaccine to be not egg adapted but adapted to the human molecules that are there. So again, it makes the vaccine have a much higher level of specificity for human protection and engendering an appropriate response. So this project, again, is one where it's about, you know, can we modify existing vaccine manufacturing processes, but tailor-make them so that they provide an outcome that's beneficial for human health. Design of macrophages, again, is another great project within this. Again, macrophages are really important in controlling the response. And as I said before, although there are maybe a small population, these small populations can have really important and significant outcomes to the end result of the disease, whether it be the appropriate response, so again, control of infection, or whether it be an inappropriate response because of incorrect signalling, these sorts of things. So approaches that use, in this case, stem cells and then being able to modulate the production of macrophages from those cells, again, give us critical information about how we can modify antigen-presenting cells so that they perform in the best way to give us the best sorts of outcomes. And so this project really drives that area and our understanding of those. And of course that has extensions into the other aspects of what we do. So if we understand what are the triggers that force the macrophage to provide a more appropriate response, we can use that knowledge for the development of other therapeutic approaches. Yeah. That's that host, I guess, directed side of the research that we have within our portfolio. And I guess we also have, as I said, the complementary side, which is about discovery and control of pathogens. I think, again, treatment of infectious disease remains very challenging, and that additional alternative approaches are required. You know, I already pointed out that there's a need for alternative approaches to antibiotic use, and also, I guess, alternative approaches to antiviral treatments as well. Just in the same way that antibacterials are suffering, antivirals also had suffered the same complications. So the development of potentially broad spectrum approaches to these is important too, and for clear reasons that are there. So we have a number of projects, and again, that are in this area, and Karen's going to specifically focus on some in the next talk from here, but I'll just talk again about a couple of these. A project by Jake here, it's about using natural proteins as synthetic protein switches. Again, this is a project that's, this has been up and running in their lab for a while, but it's only been the FSP for a short time now. But it uses modelling to develop high throughput screening approaches. It's developing, I guess, a structure around a pipeline that can give you from sequence to structure at the end there. It's very much around, being able to identify new proteins in this aspect. Also is a project by Denise, which is around the development of protein nanocages. This project, again, it uses, as it says, photodynamic therapy, so light-activated activities, to change the levels of reactive oxygen species that are there. And these reactive intermediates are critically important for being able to, they're critically important in all types of immune responses, but particularly in areas such as antibacterial, but also in cancer therapeutics. It's about trying to develop a novel protein nanocage so that you can target this activity to specific areas. And it's about, as it says again here, about using light radiation to develop this area. The project's making good progress along this year, and again here it says it's the first time that functional protein photosynthesis has been used and been loaded inside a nanocage. So again, excellent progress in this area. Just in to finish off on the last one of the viral areas, it's about DNA templated virus-like particles. Frank is here again, and he's working in this area. The project's to create a synthetic toolbox of novel virus-derived structures, and being able to use these structures to investigate the role of the geometry, the shape of these sorts of things, and how they interact, on cell recognition and uptake of these sorts of things. I think it's been known for quite some time that the host immune response uses more than just the sequence that's there for recognising targets, but it's an area that in the past has been poorly understood. This type of approach, where it's about understanding how receptors or how interactions are occurring based around the structures that are there, is a very different approach, and one that I think is challenging but really interesting. Okay, so just to sort of summarise what we've said with all of this, So our approach is really to support the production of procedures, products, etc., which help the customisation of healthcare. And that will help us with benefits around medical decisions by diagnostics, treatments, etc. that go on. It's really about supporting diagnostic testing and developing appropriate complementary therapeutics that balance the diagnostic that are there. And we're very much about providing the tools for molecular diagnostics and imaging, etc. Oops. So I just want to acknowledge our capability developers, and obviously we have a broad range of partners who work within this domain of the FSP. And then that leads me to finish up and move on to our next speakers. And first up is Karen. She's going to talk a bit about the activities that she has in with Phage.
So we are running a little bit late as we were on the sessions this morning, so I'll ask people to be concise and the speakers to try to keep to their time and not be too wordy with your question at the end.
All right, good afternoon, everybody. So yeah, I'm going to be talking about the work I've been doing for the last year. So I'm one of the FSP fellows, so I'm 1 year into a 3-year fellowship. Sorry, am I being really loud? Okay, shouting at people. So the objective really of my particular project is to try and come up with a novel synthetic biology platform basically composed of engineered and synthesized phages and components of phages to basically mitigate biofilms. So the particular target systems I'm working in at the moment are E. coli, which I'll basically be focusing this talk on. Also, I've been looking at Legionella phages and Klebsiella pneumoniae. And the idea is really to come up with this novel synthetic biology phage platform across a range of environments where biofilms can occur. So not just clinical settings, but all across like industry and the natural environment as well. So this is a nice little schematic that I've taken from a paper by the Lu Lab at MIT, where they really kind of try and see where the synergy is between synthetic biology and bacteriophage. And you can see there, phage can be used for a real myriad of applications. So I think this is really quite an exciting area for SynBio to be used in. So the actual outline of my project, so I came up with an idea to basically use a multi-pronged approach to tackle the issue of biofilms using natural phage as a template to then go on and re-engineer them or even synthesize them from scratch to make them super duper at what they do. So the idea really is to come up with engineered superphage to tackle superbugs, I guess. So yeah, so like I said, the idea is to come up with these phage-based technologies using SynBio and really to try and put Australia on the map as well, because this kind of technology is pretty well developed in the Northern Hemisphere, of course, the US and Europe, and I think it's timely now for Australia to jump on the bandwagon as well. And the idea really sort of, I guess my ultimate goal is to come up with a database or a library of these synthesized and engineered phage and their components. So the multi-pronged approach, which I'll give a little bit more detail on in a minute, is basically to use an engineering approach, also possibly to use CRISPR-Cas, which I'll give a little bit of detail at the end, and then to kind of test these synthesized and re-engineered phages using microfluidics. So the idea of scale there is to start on a smaller scale to really see how these phage will perform in terms of tackling biofilms. So I'm not going to go into too much detail, but one of the organisms I'm trying to work on in terms of controlling using phage is Legionella, and of course Legionella always hits the headlines. There is a problem at the moment in Queensland in healthcare facilities up there with Legionnaire disease and Legionella being detected in water distribution systems. So that is one of the aspects we're working on. I'm co-supervising a PhD student at UQ who's basically trying to pull phage out and trying to engineer phage. I will say Legionella is a bit of a difficult beast to work with, so it's not that cooperative, but anyway. And then the main focus of my talk today is the other system I'm working in, which is basically E. coli that are responsible for urinary tract infections, so known as UPEC for short. This is a major health burden problem globally, 150 million people affected every year. It's a huge economic burden as well. And a lot of these E. coli strains exhibit multi-drug resistance. So again, antibiotics, obviously, there's a really alarming crisis developing in terms of antibiotic resistance. And these UPEC are also responsible for anything between 80 to 90% of UTIs globally every year. And of course, yeah, the antibiotic issue is a big one, where our misuse and abuse of antibiotics has led us now to really having to look to alternatives rather than just trying to use antibiotics which aren't going to treat infections anymore. And yeah, there's a nice graph to just show how this antibiotic resistance problem has started to emerge. And the World Health Organization have said by 2050, antibiotic resistance will be responsible for 10 million deaths a year, and that outstrips mortality rates due to cancer, for example. So for my particular project, we took these E. coli host strains that the group I'm attached to at UQ, Mark Shenbury's group, at the bacterial pathogenesis lab, when we basically decided to pick these particular E. coli strains based on their K capsules. So this is the outer barrier, if you like, in these E. coli strains. So the idea was to try and see if we can isolate phage that basically attack the capsule. That's like the initial point of contact. So this is just a quick flow of how I get my wild-type phage, the natural ones, to use as the templates. It's very glamorous. You go out and collect sewage, because obviously these are all human-related phage, so that's where you start, and then just go through all these kind of traditional ways of isolating natural phage. And then the idea is to eventually come up with their sequenced genomes, use those as a template to then start the engineering process. So I performed a host range analysis on the E. coli phages that I'd isolated, and there wasn't really a nice trend. So it looked like the phage I'd isolated were not actually targeting the capsule as such. So that was a little bit, a bit confounding in terms of what are they actually targeting. But I went on and sequenced 12 of the phage, got their whole genome sequences, and that really is the starting point then for the SynBio aspect. The phages that I isolated basically turned out to be predominantly podoviruses, so this is where I get all virally nerdy. Most phages belong to this taxonomic family, the Cordoviralis, which have double-stranded DNA genomes, so most of them turned out to be podoviruses. And then I use these genomes, like I said, to be a template. So the idea is really to start looking at particular physical features of the phage to reconstruct, re-engineer. So I'll show you a little animation in a moment to illustrate really what I'm talking about. But yeah, there's things like these tail fibers, also capsids, which is basically the shell that holds the genomes. And also other things to potentially look at will be enzymes that the viruses encode for, and then also the idea is to then basically engineer them using yeast recombinering, which is originally a lovely technique that was pioneered by Paul Jaschke, who's sat here in the audience, and has been optimised by Timothy Liu's lab at MIT. Or sorry, not optimised, modified. You did a perfect job. Cool. So this is the animation that I just want to quickly show you in case anyone is not a viral nerd. I don't know if this is going to come up or not. Oh, here we go. So this is a really nice animation of how a phage targets a bacterial cell from the Michael Rosman lab at Purdue. So you can see the white head where the genome sits inside, that's the capsid. And then my particular focal point really is going to be these tail fibers. So they're really key because they attach to specific receptors on the surface of the cell And it's very much like a lock and key, so it kind of determines the specificity of the phage. And you can see once they've attached there, they can basically drill down, punch a hole in the outer surface of the cell, and through that hole they'll end up injecting their DNA. So that's kind of a nice illustration of just exactly how phage physically operate. So I was very fortunate. One of my external collaborators is Timothy Luce. I went to the Synthetic Biology Centre at MIT for 6 weeks earlier this year. And basically the idea was to immerse myself in this yeast recombinating technique under the tutelage of Sebastian Lemire there on the right. Timothy Liu is the head of that particular group and really is one of the leading pioneers with synthetic biology and phage in the world. And it's based on this paper that the Liu Lab produced a few years ago. And the idea is to take a natural phage and then to start basically switching around the different components, and in particular these tail fibres. So you can see the original phage at the top there. If you can actually start switching around the tail fibres, you can expand the host range of that natural phage. So this is kind of the basis of what I was learning at MIT. And yeah, so the yeast recombinating, if you try to engineer these phage in the actual E. coli, obviously that's going to be toxic, so the E. coli will just die and you won't be able to get your phage constructed. So instead you use yeast as the host, if you like, to actually re-engineer the phage and reboot them eventually in E. coli afterwards. So it's using a yeast artificial chromosome, and you can basically take the natural phage genome as the template and then start mixing and matching different genome components. And hopefully, like I said, you can end up rebooting the phage and get the progeny to basically form these plaques. So the little clearings on that plate there have all originated from a single virus originally. And then, like I said, this again from their paper just shows that they can end up really expanding the original natural phage host range. So that's the idea, is to kind of, like I said, make them super duper, make them even better at what they do naturally. And here, I quite like this image because it kind of shows how you go around the engineering process. So you can basically deconstruct the phage and then start stitching it back together how you want it. So taking particular features that you want to have engineered in. And again, this is a nice way of showing it. So this is, again, like that animation I showed you. You can see the tail fibres and so on, that you can actually just start switching around and doing an exchange to come up with like a better version of the natural phage. So I ended up generating 11 phage hybrids at MIT. I'm in the process of getting those genomes sequenced as well. And again, I'll be testing the actual host range on the UPEC strains I have at UQ. And this is another little exciting aspect, hopefully. Majid here in the photo has been helping to set up the biofoundry at UQ. Claudia obviously presented what that involves in terms of the robotics, and we did a quite nice little experiment which, again, talking about RSI, helped me a lot, that we were able to plate out like a 96-well plate. So you can see here, Majid used the biofoundry to actually spot all the E. coli, so this is like the equivalent of that big plaque plate that I showed before, but now on a much higher replication scale. And then I took that away and spotted my phage onto each of these little colonies. And you can see that I did it randomly, so you can see where there isn't a nice circle of bacterial colonies, the phage had actually lysed that particular colony. So this would be a really nice, if I can use the biofoundry to basically generate a lot of engineered phage in a far better, more efficient, high throughput approach. And then this is something that I would like to try. I don't know how ambitious I'm being, but basically there's been a couple of reports out in the literature where they've used phage to not actually deliver its genome into the cell, but to deliver a CRISPR-Cas system. So then you get a very targeted approach in terms of the phage able to deliver basically a gene editing system into the cells. So these are called phage mids. And that'd be something I'd like to explore potentially, if possible. And then the other approach, like I said, is actually testing this on a small scale before scaling up eventually, using like these lab-on-a-chip microfluidics, which enables you basically to manipulate a lot of parameters, also to have much higher throughput and replication. And again, the Lou Lab have done a lot of work on this in terms of testing Phage 2. There are considerations and challenges, so whenever I tell people I work on viruses, they normally completely freak out. And, you know, Ebola and HIV is the first thing that normally comes to people's mind. But in terms of this phage technology, I have this old ancient proverb in my head that my enemy's enemy is my friend. So these phage are really like the good bacteria story. These phage are good viruses. And I think as well that there could also be potential for synergy between these phage and the use of antibiotics. So there's been reports, again in the literature, that sometimes the bacteria, if they've been treated with phage, can actually become resistant to phage infection, but then there's a trade-off that they can become resensitized to antibiotics. So this again, there could be synergy in that needs more exploring, I think. CRISPR-Cas, obviously we're still learning more about just what CRISPR-Cas is and what the potential there. Like I said, the scale up with using microfluidics I think will be a powerful way of testing. And also, I'm sort of doing a bit of a science communication exercise where I'm trying to also raise that this phage therapy is a viable alternative to antibiotics, and using SynBio will make it an even more powerful tool. But that conversation is ongoing where clinicians a lot of the time aren't even aware that this research is happening. And then, of course, the CSIRO's sort of social license to operate aspects as well. Obviously, this phage therapy will need, obviously, all the legislation and an adequate framework. There'll have to be rigorous safety protocols and everything involved. And like I said there at the bottom, I put it in bold because acceptance by the wider community is going to also be key to have this research, just accelerated and maximised. And I would just like to acknowledge my mentors at UQ and CSIRO. I'm also attached, this is where I get my sewage from, the AWMC is the Advanced Water Management Centre at UQ, so they've been quite kind in taking me under their wing because I'm basically not allowed to work in the bacterial lab because of the potential for my phage getting loose, I suppose. And then my external collaborators, so Jeremy Barr's based at Monash, he's done a lot of really interesting things. He had a paper out where he basically calculated that every day in our guts we have 30 billion phage turnover. So phage are, they're busy and they're all around and inside us. And MIT as well, they were really great in basically helping me get to where I am now with being able to engineer these phage at UQ. And with that, I'd just like to say thank you for your attention and if you've got any questions.
Can you engineer like a phage that will, with like it's a multiplex legs, so you can target many bacteria at once?
Yeah, that's a really good question. That's exactly what I'm aiming to do. So you can, so some phage naturally will have different, what we call tail spikes, or these fibres on the end. And they can switch depending on which cell type they encounter. So they can almost sense, oh, this cell's got this particular receptor, so I'll use this tail fibre to attach, or vice versa, whatever else they've got available. So yeah, that's definitely one of the things I'm going to be doing in terms of engineering is, putting on different tail fibres so that they can, attach to different cells.
How do you plan to deliver the phages to the target site?
So you basically would have to end up with a high enough titer stock, and then, on the microfluidic side, that would be easy because you can just inject it. On the scale up, I don't know. I think that's where eventually I'll have to talk to real engineers, if you see what I mean. Like with the Legionella project, that eventually is going to be applicable in water distribution systems, which again, I don't know myself because I don't know about plumbing and engineering, but in the early stages it's going to be easier on these lab-on-a-chip devices.
I have a question. So there's a Navy always has a problem of a biofilm growing in their boats, and they put trying to get rid of it. So can you make a page with a phage? You know, the big bacteria starts drilling in, you know, they get annihilated because the phage is not alive technically.
Yeah, that's possible. That's possible. I mean, the other thing is, I don't think. So that I also have this other anecdote where I don't think people realise, like for things like certain foodstuffs like cheese, that's already been sprayed with listeria phage before it gets packaged and sold for consumption. So yeah, there's all sorts of ways that phage can be delivered, and like you said, if it was a paint or a spray or something, but yeah.
So other than the host specificity, do you think there are any other aspects of infection that you could optimize with synthetic biology, or is evolution kind of got it pretty well right already?
No, I think you're right. So that's one of the other things that I briefly showed on the slide is if you had virally encoded enzymes. So again, Timothy Liu's lab has that where they engineered phage to release dispersins to break up a biofilm. So there's definitely other aspects, not just the physical kind of components of the phage itself. You could definitely engineer them to deliver an enzyme to do something like that, like control the biofilm that way.
So there's a long history of phage use in medicine. Thinking ahead, if bacteria are good at anything, it is getting around specific phages, or phage, I guess. So what are your thoughts about how you put all of this effort into engineering a phage to solve a particular problem, and then right away a new strain of bacteria pops up that has circumvented your best efforts, and you're just not going to be capable of doing the number of iterations it takes to keep ahead of the organism?
Yeah, so you're right. So I guess an ongoing arms race in terms of the evolution of the phage and its host. But I think that's probably the powerful tool that synthetic biology can offer. So I think that's, yeah, you're right in some ways, but I think that's where SynBio comes into its own then, enabling us to try and work around what naturally happens in terms of the evolution. So, yeah.
Okay, one last one, and then Elliot.
The concept of the biofilm, so my understanding is that biofilms is a sort of extracellular matrix of, you know, glycans and proteins and things. And actually the bacteria part of it is kind of slightly superfluous once the biofilm is being formed. So because it's a structural matrix, so one has to try and remove, dissolve, you know. So I'm just trying to work out what is this, you know, what the phage concept allows you to do apart from delivering something. You know, I'm just trying to work that out. Also, all the bacteria are actually immersed below the biofilm, but it's just a massive matrix of things.
Yeah, so that's one of the answers I just gave to Tom, is that you could also engineer the phage to release enzymes that will help break down that extra cellular matrix. Well, if you had the phage to engineer to release a dispersin and you start breaking down that matrix, then that makes it easier for them. Maybe you could then add in, because you'd make a phage cocktail anyway, so it would be more than one type of phage. So you could then have one that releases an enzyme to break down the matrix and another one to go in and target the particular bacterial host you don't want in the biofilm.
Thanks very much, Karen.
Hi everybody, my name's Elliot. It's really cool to be here and get a chance to talk about my work down at Army in Melbourne. Really feels like a change of topic based on all the things we've heard today, but yeah, let's see where it goes. So really the you know, this is the title of the fellowship, the 3-year program that we put together. But we're about 5 months into this, and sort of it's kind of split into 3 very concise arms. With really the control of reprogramming in stem cells being kind of the final event of the fellowships. Really, what I wanted to talk to you guys today is really about our first part, which is kind of the base camp for the whole project, which is the creation of this optogenetic toolbox, which eventually we want to use to control stem cell differentiation. A little bit about me. So I've also only been in Australia for 6 months. I moved here from Manchester, so the weather here is making me feel quite at home. And really, I specialise in sort of molecular function of animal opsins, so light-sensitive proteins. And did most of this work in jellyfish, box jellyfish eyes actually, but then quickly realised I needed to sort of get more into a field that had more application to human health, and that's really where I sort of came into the synthetic biology field. Today, I just really want to, I imagine many people here haven't heard of optogenetics at all, so I really want to give a sort of introduction on that and protein-protein interactions, and then actually sort of talk about the progress we've had with this arm of the project, which has already worked pretty well and pretty quickly. We've got up and running, so hopefully show you guys that, and then where we're going to go after that. So, optogenetics, I'm going to just assume I'm talking to people who haven't even heard the word before, but it's this new field emerged. The tools have been around for a while, but really as a field hasn't emerged since about 2005. And really it was this idea that we're taking light-sensitive components in nature and repackaging them and using them in mammalian cells or even whole organisms to control cell behavior with light. So this is one of the first photoreceptors that was isolated from algae, and what's great about these photosensitive proteins is that they induce hyperpolarization or depolarization of the cell membrane, depending on which specific protein you're using. So if you take the DNA out of this organism that encodes that protein and you give it into a neuron, you can control that neuronal firing with light. So in response to light, you get depolarization, and the neuron turns on, or vice versa, it turns off. And really, what it does to the neuron is very much dependent on the protein you're using. And as you can imagine, as the field has grown, we now have a whole library of not only natural variants, but optimized variants that do different things. And now there's such to the point where you can put these tools in vivo and have an awake behaving mouse where you can control neurons in real time and see the impact of circuits being modified in real time on behavior. So it really is an awesome, really cool part of neuroscience. So really, all it is replacing the neurotransmitter part with light. And why would you want to do that at all? Apart from it being well, cool on the surface at least, it's the next generation of replacing traditional pharmacology approaches. And I think all of you here could appreciate the fact that light is faster. And if you've got a tissue that's not sensitive to light, such as the brain, and you only express your protein in specific places, you can illuminate the whole tissue, and only your cell of interest is going to be activated. So you get this unprecedented spatial control, as well as the unprecedented temporal control of neuronal firing. So if you haven't heard of optogenetics before, it's a super, super powerful group of technologies, and it really is only sort of been used in the last 10, 15 years. And sort of it has both therapeutic and kind of business applications, because it's not just about learning how these circuits work, but then also can we actually manipulate them for a therapeutic benefit. But having said all of that, that's nothing really about what I do. So really, this is this is really how the field is established. These are called ionotropic tools. They control membrane depolarization. I don't really care about that. What I care about is controlling other parts of cell behavior. So cell behavior isn't really just about depolarization or hyperpolarization. There are many other signaling pathways that many of you will know about that are controlled by sort of non-electronic processes. So really my PhD was all about optimizing metabotropic tools. This is the idea of G-protein signaling, which we don't need to go into, but is really one of the main modalities by which cells communicate extracellular signals. But really what this project is about is going into the protein-protein interaction space. So again, I probably don't need to tell you that many processes inside the cell rely on protein-protein interactions, and I'll show a few examples here. And really controlling these processes with light is where we want this project to sort of excel. So it gets into a bit of alphabet soup when you start going through all the different receptors available for this, but really the point of this slide is that they're already in nature many different proteins that naturally exist that do these kind of protein-protein interactions in response to light, and not just one wavelength of light, but different wavelengths. So here you've got the 5-PIF system for heterodimerization, you've got homodimerization, you get the idea. We've got this slew of potential tools we could be using to do this, but at the moment the real difficulty is that if you want to apply these to your novel problems, say a protein-protein interaction in the cell that you don't know what it does, and you want to interfere with it in an objective way, trying to sort through the various tools available and know which one to use is very difficult process, takes people a lot of time. As well as not being particularly advanced in terms of the tools themselves. It's really this is one of the newer arms of an already pretty new field. So really the first aim, or the first arm of this project, was to create a kind of centralized platform for taking target proteins and, really aimed at researchers that don't work with these kind of proteins, and making them light sensitive in a sort of organized, efficient way. So this is this idea of this OVC, this optogenetic vector collection, that we want to use to control protein-protein interactions. And so this idea that instead of having specialized receptors for specialized sort of outcomes, we sort of wanted this central system that we could apply to all different types of protein-protein interactions inside the cell. And these are some examples of the kind of interactions that we want to control. So I won't. There's not really too much to take home from these slides, but in terms of how we wanted, I've kind of already said it. But the way in which this approach differs from other approaches is really we wanted to create a vector library that already had the photoreceptors in place, so that you could then take your target gene, and that's all you needed to know. And then we basically would have the system that could then be, in one or a few cloning steps, be put into the optogenetic vector. And really, the last 5 months has been a lot of cloning, and I could have put a lot of boring slides up here of me pipetting, but what we've come up with is basically this library of photoreceptors in different orientations, and also room in the middle for different linkers. And you can use things like P2A peptides to ensure an equal ratio of proteins inside the cell, if that's what you particularly are interested in. And you can also use these sites for fluorescent tags. And really there's a lot of flexibility in there in order to sort of provide researchers with the best possible base point in which to tackle their optogenetic problem. So yeah, exactly. So you know your insert. And really and in terms of, for those of you who are into your restriction enzyme cloning, really the kind of trick, I guess, that we used is by having compatible insertion sites so that we can reduce the number of cloning steps in order to put in your target gene into each orientation in parallel. So this is the lineup of kind of the first generation of constructs that we've produced. And by produced, I mean this is sort of we've harvested from what's already available in nature, and with these the ones that we've sort of centralized into our platform. And what's cool is that they cover a bunch of different types of protein-protein interaction, but also to different colors of light. And if you do different colors of light, that opens up the possibility of using them in tandem. So really, with these 6 tools, you could already do quite a lot. So really, the sort of what I want to show now is just a little test system that we used at the end to sort of just show, and this is we've kind of got through a couple of rounds of it, and I want to show you some data from that. But really a good test system we thought was caspase 9, because this is a protein that exists in cells that naturally, so it's the initiator caspase that induces cell death, but it does that through critically a homodimerization reaction. And we thought this is an easy system in which we can test our vector library. So it has been done in the past, and it's been used in various safety switches for T-cell therapy, but we thought we can maybe optimize what's already out there, as well as sort of actually providing that kind of a control plasmid in our collection already, because this is quite a good thing to have in there with it. So what we did, so we've got our caspase 9 produced, and then we've just basically put that into our 6 different types of photoreceptor in both orientations to see if there was a particular difference between putting it in on the N or C terminus. And then we did cell viability assays just to see what happens when we put these in the cells. So these boring charts are basically just showing that in the dark, putting these photoreceptors in doesn't give you any effect on cell death, which is good, because that would be a problem. So in the dark, there's no dark activity. But then when we turn the lights on, in the blue bars, in a few of our constructs, we saw the significant cell death, which is what we're looking for. We got a couple of interesting results here with the RSLP, where we actually managed to make cells healthier somehow. Haven't quite figured out quite how we did that, but we think that might just be sort of an artifact of our experiments. This is really done in sort of the last few weeks. But critically, we've got constructs that we believe we can control cell death with in a rapid and efficient way. So this takes about we leave, for those who sort of don't do optogenetics things, we leave these cells for 6 hours under kind of moderate blue light, sort of no stronger really than room light, but specific to blue, which is what most of these constructs are sensitive to. So it works. And then we took the constructs that worked, the AU1 and was it the CRY, and put those through flow cytometry to sort of actually really assess, okay, are we killing these cells or are they necrotic? And what this showed was that we were indeed pushing cells into apoptosis rather than pathways that would be not controlled by cell death. Because if, for example, we were just frying the cells with light, etc. And this in early stages seems to be backing up that we're doing something specific to the cells. Okay. So really, that's where we're at with this arm of the project, and I know it's kind of only a sort of small section of hopefully where the 3-year project will go. But I kind of wanted to show more of sort of one part of it rather than just talk about what we're potentially going to do. So we're sort of expanding the library a little bit to include some more photoreceptors, different linkers. We're going to test those out, but hopefully get this arm of the project kind of wrapped up soon, and we'll actually start using it for the main aim of the project, which is this reprogramming of stem cells, and that's really where, hopefully, if I do this time next year, there'll be much more kind of advanced sort of in that area. And yeah, that's really the thing. So we have, is there anything else? Yeah, so the second arm, which I really didn't mention, but it's kind of in tandem with the first arm, is this idea of we're actually going to build new illumination devices, so to go with this library, so that we have this paired system of the actual constructs and the hardware, so that you can have a whole optogenetic setup kind of packaged in one sort of setup. So hopefully we're going to start testing those in the new year. And with that, I want to acknowledge, the photo, I thought it was a joke, and then they actually did it. So this is it. They've got some Reservoir Dogs fantasy going on, and I just went along with it. But the lab's been really great. I've been there 6 months and I'm having a great time, and particularly thanks to Alex, who helped a lot with the cloning, and to Harold and Andy, my supervisors. And yeah, thanks.
So you showed that you got about 50% apoptosis with your system. What do you attribute? So is that a good result for a safety switch, or is that a poor result? And is it to do with the optogenetic control, and you get something better or worse with other systems?
Yeah, I think obviously 50% in some medical applications is not going to cut it. I think in this system it really has a lot to do, I think, with the transfection efficiency. I think it's just the fact that we're just doing transient transfections, just throwing the DNA on there, and including throwing the reporter on there as well, so we're really only assaying cells that have taken up both, whereas you've got a lot of cells that will take up one or neither. So I think it'd be more on, the onus would be on the people, the application, making sure that your expression is good, is robust, and that's really where you need it. So
if you had reporters that would allow you to sort cells that had both the optogenetic system and the reporter,
yeah,
then you think you'd get a much more efficient.
Yeah, I think that's part of this sort of second generation we're going into, is actually incorporating fluorescent tags into that same construct. So you have your optocaspase 9, and you also have a tag in there. So as you said, you can actually then pre-select the cells that you actually want. But you're right, for those medical applications, it really, it's very important to know exactly where it's expressed. Otherwise, you're going to get in problems.
And then with conventional Cas9 systems, how efficient is their killing?
I don't know off the top of my head a number to give you. It depends on there are systems though that do. But.
I have a related question. So, on the level of your protein components, so what is actually dynamic ranges that you're achieving? Meaning, plus minus activation. What is the maximum activity range? Because on transfected cells, you get a twofold increase. Your control with
FPVP, yeah.
FPVP is around 20-fold.
Mm-hmm.
But whether it comes from transfection, it actually comes from the modulation of the basal activity where you switch.
Yeah.
Have you tried to resolve that, or?
I mean, so the more detailed data maybe for that would be the dose response curves to light maybe, and you're talking literally about what is that range you can actually get to, and whether it's better for different tools. I think this is maybe where the linker domains come in, whether maybe we can try and, I think that's an area where we can try and optimize because that dynamic range could be improved. I'm not sure that's a proper answer to the question, but there are ways we can improve it. But yeah, not sure I quite answered that.
Oh yeah, well, I had a similar question. So, can you regulate the response by changing the intensity of the light source? Or,
yeah, definitely. So there's, I probably should have put it there, but there's nice dose response with light. So as you improve the light, there's an increase in activity. So, and nothing in the dark, importantly.
Tissue penetration can be quite limited. So you think they give you a issue for deeper applications?
Yeah, definitely. I mean, it's a limitation of optogenetics in general, and this is why we incorporated red light sensitive tools, because red light has this advantage of transversing tissues with less interference. So, but these tools are have a less impact. So it's what's the right way to say it? They're basically not as powerful. It's a trade-off. But if really deep tissue penetration was what you were after, you would use something sensitive to red light as opposed to blue light. Or you'd have to go down more of a hardware route of how do you actually transduce those areas of interest. So a lot of the ionotropic optogenetics is all about optical fibers and implants and micro LEDs and things like this. So there are two ways around it.
Ready? Okay, fantastic. Thank you very much. Okay, next one is Layton. I know I'll fix up these slides.
While Andy's just bringing that up. So I'm from the ARL site in Geelong, CSRO. So Karen mentioned that people think she works with Ebola sometimes, but we actually do. So this is the site where you can do PC4 work right up to things like Ebola. But today I'm going to be talking to you about SynBio strategies to combat dengue infection. But what I'd really like to sort of say is that underlying this is a really important technology that allows us to work in non-model species in the genome-wide scale, which you can't do unless you're working in human or mouse cells. And so I guess at the end of this talk, hopefully you can pull out of that this platform that we're developing is really important for other projects as well. So dengue, as a lot of you would have heard, it's a big problem worldwide, especially in sort of low socioeconomic areas. You can see here the data from the WHO that dengue infections are on the rise, a really rapid rise over the last 50 years. This may be partly because we're testing more for it, but certainly there's a lot more spread of dengue, and that's due to climate change and movement of people and things like that. So you get up to nearly 400 million infections a year, with 500,000 of those being hospitalisations and 20,000 deaths. So it's quite a big problem. If we're going to develop new strategies to help combat dengue, one thing we really want to look at is, I guess, how dengue spreads. And you would have probably heard that, mosquitoes spread it to humans, then non-infected mosquitoes get it from humans and spread it to the next human. So I guess to really understand this sort of cycling, we want to look at how the host deals with the virus and why mosquitoes don't succumb to the virus and humans do. And I guess what we're really trying to find is what circuitry or what pathway can we block or interfere with to either help the human immune system deal with the dengue infection, so as a therapeutic, or potentially even change the mosquitoes so they can no longer harbour the virus and break that cycle. Unfortunately, in this space, the host response is, there's not a lot of information out there, but with the, I guess, invention of CRISPR-Cas9 and loss of function studies, we can certainly start looking at different mechanisms as to, I guess, how the host or the vector immune system does deal with the viral infection, but it really does take too long. So if you're trying to do this on a gene-by-gene basis, it's quite a long process. So I guess the platform technology that I was alluding to before is called GECKO, so it's genome-wide CRISPR knockout screens, and this essentially can do a loss of function study on every gene in the genome. So I'll walk you through this fairly quickly, considering how complex the process is. You start off needing a library of CRISPR guides. So ideally you want to design up to 3 or 4 guides per gene so that you've got good coverage, and you want that to every coding gene in the genome. This library then gets inserted into a lentiviral vector system, and using, I guess, the appropriate virology, you can insert that lentivirus into the cells of interest, but only with one lentiviral vector going in per cell. So you never get knockout of more than one gene per cell. But obviously you need to do this on the hundreds of millions of cell scale so that you can cover the whole genome. Then I guess, depending on your purpose, you design an assay that will, I guess, give you your winners and losers in terms of what genes do what. So for us, this is going to be an infection assay. And then from the winners and losers, you can do next generation sequencing to see which gene was missing to cause the change in phenotype. So I guess all of that summed up, what we're really trying to do in our project here, create a pool of mosquito cells that in total are missing the entire genome, but only one gene per cell. Then you can see in green here, we're hoping to see if we get the loss of an antiviral gene, we'll get more virus produced. If we get the loss of a proviral gene, we'll inhibit virus production. And so you can see, for example, if we were to find what genes are proviral in a mosquito and we're able to remove those from the mosquito population, they'd no longer be able to harbour the virus and give it to us. The screen that we're trying to develop to, I guess, deconvolute this pooled screen is flow cytometry. So we can take the cells, look for viral antigen levels, and then sort them using a fax machine, and then move on to our NGS. And the ultimate product here is going to be a mosquito with either an antiviral gene that's enhanced so it can fight off the disease, or a proviral gene removed so that it can no longer harbour the disease. But I guess as sort of offshoots from some of this work, we'll definitely be looking at things that we might be able to trigger in the human immune response to help us fight the disease better, so therapeutics. Very briefly, I'll just take you through our aims. So we want to initially develop a gecko library for mosquitoes. A lot of people get confused when I say this. Why are you doing a lizard library on mosquitoes? So genome-wide CRISPR knockout library for mosquitoes. Then we need to design this assay. So at the start of this project, it was all just a concept that we might be able to pull apart this convoluted screen and deconvolute it, then undertake the screen and essentially make a final product. And in this case, it was probably going to be a cell line that was missing a gene or had a gene enhanced, and see if we can change how much virus is growing in that cell. So this is, I like pictures. This is a far easier way to look at all of this. So the first step is designing the library against mosquitoes. So we did this in collaboration with another group at CSIRO, and we've designed a CRISPR library that is targeting, it's got greater than 34,000 CRISPR guides targeting over 10,000 mosquito genes, and for more than half of these genes we've got at least 3 guides targeting every gene. And this was designed using an in-house algorithm that Dennis Bauer's group have now published. So we've now had those designed, and we've synthesised the oligos that would then be inserted into the library. So I guess we're up to this sort of library cloning and generation where we run into a pretty big hurdle. So the initial backbone that's used for the gecko system was designed by the Zhang lab, and so we got the vectors from them. Unfortunately, these mammalian promoters, so you initially need to make a cell line producing Cas9, stably expressing Cas9, and then put in the guide library. Unfortunately, none of these promoters work in mosquitoes. So we kind of had to go back to the drawing board a little bit. So we had these 2 human lentiviral plasmids, and we needed to, I guess, mosquitoify them. So we started with the Cas9 first, and this was a pretty straightforward process. We synthesised a mosquito promoter, the OPIE2, and just using basic cloning, we were able to cut out the EF1A promoter and clone in our OPIE2. So we've now got this plasmid ready to go, and we're now in the process of creating a mosquito-stable Cas9 cell line. The guide library, which comes in the second vector system, is a little more complex. So we had to go through a little bit more of a process where we synthesised our promoter as well as the puromycin that's used for selection, because there was no other way to essentially clone these in frame to each other. So we created a larger fragment, and this couldn't actually be synthesised because of its complexity and length. Then once we had that fragment, we were able to cut out from the lentipuro the original promoter and the puromycin and put in our mosquito promoter. But this still left it half human, half mosquito because we had the U6, which also doesn't work for mosquitoes. So our final step here was to clone in mosquito U6 that we know works in our mosquito cells. So we finally had the appropriate backbone now to go and make our library. So that kind of set us back a little bit, but we have the core backbone there now, and this is, I guess, an important resource for other people doing mosquito research. So the next steps for us, we need to generate that stable Cas9 expressing cell, like I said, and then we want to insert some guide RNAs that we know work for mosquitoes into our new backbone and just validate that and make sure that it works and that we can get knockout before we go and put in the whole 34,000 guides. So this is kind of where we're up to. This is underway and I think we've made a lot of good progress in a short time. But what we wanted to do in the meantime was skip forward to start developing this screening assay, because essentially if you've got a pool of hundreds of millions of cells and some of them are growing more virus and some aren't growing any virus, you do need a way to isolate those cells to find out how they got that phenotype. So we've learned a lot of lessons on this path as well. So one thing that was really important, I guess, we needed to develop an intracellular staining, so the virus infects, goes inside the cell, reproduces itself and comes out. So we've developed the, I guess, intracellular antigen detection, titrated our antibodies, and worked on virus inactivation methods because we need to take these out of the class 2 before we can start running them on flow cytometers, and you really don't want them still infectious when you do that, especially if you were doing Ebola. So one of the other things we came to learn very quickly was that we needed less than one infection cycle for this to be functional. If we let the cells, I guess, be infected for greater than an infection cycle, the whole population started to have different levels of infection. And essentially that's what our readout is. We needed a single level infection. I might just move up. So what we're aiming for here was this sort of blue graph here, for those of you who do know flow cytometry, and it's not an easy thing to get, just essentially all of the cells infected with the exact same amount. So to do that, we had to do these virus cycle studies where we infect for a period of time, so in this case it was up to 8 hours, and by the 12-hour mark the virus was starting to be released from the cell. So as long as we did our infections for less than one virus cycle, we had a very uniform amount of virus infection to work with. And the last thing was how much virus do we put in? And we also found, I guess, only by putting in very high titers, so we work in something called a multiplicity of infection, only by working with 5 viral particles to every cell did we get that really nice uniform amount of infection that we could then use to sort from. So essentially this is our facts here. For those of you who haven't done facts before, along the bottom here is our antigen. This is our negative control, so you can see that we're essentially setting these boxes up to say anything above that box is positive, and then we used a wild type population of cells that were infected with virus to say anything above this population also had an altered phenotype. So what you're seeing here is this is our wild type cells. Anything that sits in this box essentially would be proviral. Anything that sits in this box would be antiviral. So you can see just by chance a small number of cells do fall in those boxes even without doing our gecko screen. And using a different library, because like I said we haven't got our mosquito one up and running yet, with a wild type here you can see a big change in the proportion of cells that are negative for virus and a big change in the proportion of cells that are positive, that have a high amount of virus. So essentially what we're seeing here is because of the gene knockout, we're getting an overrepresentation of negative cells and an overrepresentation of high virus expressing cells. We then sort these into different populations, so you can see we get just negatives. In this case, when we hit our fluorochrome with the laser, it actually loses some sensitivity, but we do believe we sorted only high virus expressing cells in this, and this is now off for next generation sequencing. So from here we should get a list of proviral genes and antiviral genes. We've done this in triplicate, and essentially once we do the bioinformatics we can then go in and validate some of those gene targets. So all in all, I guess we've got the workings of our flow cytometry assay and we've got this gecko screening up and running. Gotten over a few roadblocks in trying to work in the mosquito system, but hopefully by this time next year I'll be having the gene list for pro and antiviral genes for mosquitoes. And yeah, I guess just like to thank all the people in our lab. Again, got quite a big lab, and Coslin and Andrew work closely with me on this project, and Dennis Bowers' group and Lawrence Wilson helps a lot with our bioinformatics. Thank you.
Thank you.
This virus evolves so fast, and you plan like to then stack mutants that might have like a parallel, effect, so that it's more difficult to overcome that virus.
Yep, absolutely. So I think something like dengue doesn't mutate as quickly as some other viruses. So, but definitely you might look at a multi-pronged approach. I think when we're doing our validations, we'll certainly try and understand the mechanism of each target individually. But then if we do try and make a mosquito line that is virus resistant, it could take 2, 3, 4 different gene deletions.
Maybe a silly question, but why did you use the virus that can replicate on its own? Well, because you run through the different infection cycles, why you just get one that it can create a fully functional copy of its own?
So I guess the whole point of this is to look at the host response. So the only reason we're looking at a single infection cycle is so that in those cells we, I guess, don't confuse cells that have been infected with negative cells. So we really want just a single point of infection, a single mass of antigen in there. Whereas we definitely want the virus replicating in the cells because that's the natural state that it's going to get into us.
Yeah, thanks. That's awesome talk. I was just wondering, could you maybe look at like a gain of function approach down the track where you might, the loss of functions, that's obvious way to start, but...
Yeah, so I guess the parallel study to this is the SAM vectors. So they're activators of each gene, and absolutely would love to do that, but it's a big process just remaking this backbone, so we'll have to talk to Claudia about the next project.
So how are you measuring the virus inside of the cell in order to determine?
Yeah, so we're measuring viral antigen with an antibody.
Right. So are you measuring one particular antigen?
Yep. I guess we've gone through a process of looking at 2 or 3 different antigens, and what we've found is that the antigen and timing need to be matched because they do produce at different times, but it's not necessarily restricted to an antigen, this kind of data.
Well, because I'm wondering is, it could be possible that you're measuring an antigen that appears to, that particular antigen might be at some wild type level. However, that cell isn't producing any functional virions, right? Or it's not producing some other protein at all.
Yep. Yeah, you're spot on. So the gecko screen is, I guess, a little bit prone to false negatives, but pulling out the positives will at least give us the mechanisms. And at this stage, we just need a set of genes that we can target. So yeah, we might miss some, and it's not until we do the validation and actually look at live virus that we'll know if we even need to validate. But yeah, so in this case, yeah, we might have some false negatives.
Can the mosquitoes do vertical transmission of dengue?
It's a good question. I don't think so. No, because they do it on a blood feed. So I'm pretty sure it has to be horizontal to us.
So there's no good way of doing an experiment to know if dengue-infected mosquitoes were just a smidge more fit than non-dengue-infected mosquitoes.
Yeah, I'm not sure. I couldn't answer that one for you.
And then so is your idea to make gene knockouts and then produce mosquitoes with those gene knockouts?
Yeah, ideally, I think as part of this project, we want to get to a gene knockout cell. But if we have time, definitely we've got the capacity in our receptory et al. To make a gene knockout mosquito. And I guess we'd like to even have them have a blood feed on dengue-infected blood and see if they can resist the infection or not get infected if it's not a resistance.
Just following on from that, I mean, there are gene drive technologies, Oxytex technology seems to work very well. So have you thought about it?
Yeah, so one of the ultimate goals for this could be using gene drive to, for example, remove a receptor. If there was a key virus receptor that we could identify and remove from the mosquitoes, and we can still have the mosquito population, so they still play a role in the ecosystem, etc. But the only impacts that can't hold the dengue virus, that would be the ultimate goal. I think we like to set some little goals along the way, for example, like understanding how we might get therapeutics or things like that, because obviously it's a pretty big goal.
you expect different targets, that you find different targets with the different dengue serotypes?
Yeah, it's a good question. We're at this stage only planning on working with a single serotype, and then in our validation step, we'll work with all 4 serotypes. So yeah, it's absolutely possible. Ideally, we're looking for targets that only cover all 4. So in our validation, if we come up with, let's say, 20 or 30 different targets that are proviral and antiviral, the ones that validate across all 4 serotypes will be our lead candidate. That's a very good question.
Okay, that's it. Thanks very much, Dan. Yep. Outside?
Afternoon tea, just outside.
Okay, does everyone get that? That closes this session for us. Thanks very much for attending. Afternoon tea outside here, and please make your way across the road to have a look at the posters. Oh, no? Okay, you don't have time for that, but if you haven't put your poster up.
If you can get back in half an hour and be here.
So the quick ones among us are allowed to go across the road. Check as you cross the road, look both ways.
So my name is Aditi, and let me know if it is too loud in the back. So I am the Maximising Impact Application Domain Leader. So this session will obviously focus on some of the updates from our application domain. And we're a little different from some of the other application domains. Like Andy and Health and Medicine, we only started at the beginning of this year as well. So in that time, it has been a steep learning curve about synthetic biology and how social science plays a role within that world. So I'll just give you a rundown of our team. So I'm a psychological scientist, so my work really revolves around cognitive and behavioural factors associated with, uptake of novel technologies. So much of my work has been in the water resource management space, as well as biosecurity. Then we have Lucy Carter, who has expertise in institutional analyses, governance and bioethics. Do you want to do a wave? You'll come up later anyway. And a lot of work in agricultural systems. Then we've got Liz. Liz Hobmann is also a psychological scientist, and she's done a lot of work in behavioural economics, looking at the effects of messaging in energy use and the finance sector. And then Alison is up the front, so maybe some of you saw her at the Yeast Conference, but she's a specialist in IP and synthetic biology based at the Uni of Canberra. So our research is conducted predominantly using in-house expertise within CSIRO, with the exception of Alison, who's our FSP grant recipient. We do plan to scale out our capability moving forward, hopefully through the FSP fellow process, but for the moment it's just us. So another kind of key difference that much of our work is not really discrete sets of projects, but it's kind of a rolling process where each stage of our research informs the next, because we really need to orient ourselves in this space before we kind of charge ahead and make kind of social science decisions. So just to give you a rundown of today, each of us will present on a different research activity that we've conducted. So I'll provide a general update and some insights into our public focus group study. Lucy will then speak about her qualitative case study examinations of 2 possible SynBio technologies and their complex stakeholder environments. Liz will provide an early peek into our national baseline survey, a survey which Liz and I designed with the help of some of our FSP project leaders. So you know who you are in here. And finally, Alison, she came on board in August, and she'll present her early thinking on the legal implications of SynBio technology in Australia. And then tomorrow we'll have a spillover session where we would like to hold a general discussion with all of you about the possible ways or pathways for maximising the impact of SynBio science through the utilisation and co-planning of biosocial science. So this is an important conversation for all of us to have as a collective, because it not only speaks to our key FSP indicators that Claudia spoke about, but the need to co-develop future SynBio science, which is a really truly innovative aspect of this FSP as well. So I'll just give a timeline check of where we're at. So our research is divided into two broad methodological processes, the qualitative and quantitative. So early in the year, we began our process of biosocial engagement and conducted workshops with the application domain project leaders to orient our application domain, because we'd never really worked in this space before, and open that line of communication between biophysical scientists and social scientists. All of the participants were very generous with their contributions, despite this being outside many of your comfort zones, which we were really pleased about. And the sessions, I think, went really well. So during those workshops, the application domain project leaders prepared different stakeholder and actor maps, which Lucy will kind of cover a bit of, which Lucy transcribed into more formal maps and distributed these to the application domain participants for their reference and use. We then used these mapping tools to initiate our case study identification, and then Lucy began her case study analyses using a variety of data collection methods. In the meantime, we also started developing our national survey, and we conducted public focus groups to trial elements of our survey to ensure that our survey content was ground truthed in the public domain. So after an extended lead time, to ensure that our SynBio scenarios were making sense and our surveys were on point, so asking the kind of questions that people might ask in the real world and make sure that everything in the surveys made sense. And also to liaise with comms, just in case there was an onslaught of media inquiries about some of the technologies we were putting out into the world. It took a while to kind of go through that process. And so we finally released the survey into the public last week. So it's still very much in the field, but Liz will kind of give a bit of an update on that. So now I'll kind of move on to the public focus groups. So these happened in Brisbane, and there were 7 different focus groups, and each of these were across 3 different sessions, So each of these were in 3 different sessions, and some sessions were divided into 2 focus groups and some in 3. And each little group were given 2 scenarios. So the scenarios that we covered, based on the science within the SynBio FSP, were related to the culling of chicks in the chicken meat industry, bioremediation using pseudo-organisms, natural fibres with Maddie's group, mozzies with Dan's group. What else have we got? Oh yeah, conservation, so genetic engineering for conservation with Owen's group, as well as invasive species management. And the reef, so coral. So in those small group activities, people discussed one technology for about 25 minutes together, and then, so they were asked to think about the risks, the benefits, some of the deal breakers that they might have with respect to this technology, and some of the questions that they would want to ask scientists. After that, they were asked to kind of synthesize that information and present it to the broader group. So it would require some level of comprehension of the technology to make sure they were really understanding what the messaging was. And so they did that for 2 different technologies each. So these are some of the broad cross-cutting themes that came out. So you'll note that there were 7 different technologies that we talked about. So there's not one size fits all in terms of some of the themes that came out. But generally, participants thought these technologies sounded like a viable solution to some of the problems we have in Australia. They thought some tech could restore biodiversity while others could reduce pollution and waste. A dominant theme was, of course, the discussion about human and animal health or safety across all the technologies we talked about, and the risk of dual use, which came predominantly from the fear that the tech might be sold internationally and we, Australia, would lose control of it. Gaining a national economic benefit with these Australian technologies was really important to people, and there was a strong sense of national ownership and fostering commercialisation opportunities if they were present for the technology. But overall, maintaining control within Australia was really important. And broadly, people were keen to ensure that access to the technology was fair across the different end users, and that technology that was kind of put out into the world had a very simple governance structure. So some of the things we tested was related to, possible government regulatory requirements for the technology, and for I guess maybe mozzies or for the conservation technologies, there was a whole host of different regulatory bodies. And I think that really freaked people out because they're like, oh God, our government can't work now. How's it going to govern these technologies in an efficient way? So I'll present 3 of the 7 scenarios just for ease of time. And also we'll be presenting the slideshow scenarios. So I'll ask if you please don't take a picture because they're in the field at the moment. And yeah, it's just easier for our lives if the pictures aren't out there. So this is the slideshow or slide set that we gave to people related to engineering for conservation or synthetic biology for conservation. So it starts off just by framing the problem. Don't know why I'm going over there, I can look here. So it starts off by saying, this is the problem, animals are endangered or extinct, and that's an environmental issue. Currently, these are the ways in which we control endangered species, but of course the technology, the current manual methods are not keeping up with the rate of the problem. So then with new synthetic biology technology, it would be possible to recover endangered species by reintroducing genetic diversity. So this one, it was easier to kind of talk about synthetic biology in the context of bringing back species if we talked about genetic diversity. And that was Owen's kind of idea, because it is quite an out there topic for many people to say, okay, this species is extinct, we could bring it back. But if we talk about kind of creating more diversity in that genetic pool, it's a lot easier for people to stomach. So that was the information they were presented with. And then some of the themes to emerge. So for conservation, some of the benefits were, of course, restoring biodiversity, because that's how it was framed. So you would hope that people interpreted that this is a really good thing to come out of this technology. Reducing the chance of a species to reach extinction. And Australia is, of course, leading the way in this type of environmental solution. Some of the risks that people highlighted. Will the species change? And so this was a big one, and it came up a few times in some of the different technologies. So people were okay with some sort of genetic manipulation or synthetic biology application as long as the things that they knew remained the same. So with the eggs, for example, as long as there was no difference in appearance or taste of the egg, people were a lot more on board with that idea. And similarly in this context, if you are changing the DNA of a species, it should still look the same so that it's familiar, but it would be risky if it changed in appearance. Unexpected impacts of the organism in the wild. And this is, this idea of uncertainty and the unknown, which I think this type of science inherently has from the public's perspective, and probably from yours too. And abuse of the technology. So this idea that, if we can bring back species, what are they going to bring back? What are they going to use them for? some of the key deal breakers for this technology. So people were really not okay with this technology if it was used outside the boundaries of its intended use. So if it was to bring back a very functional animal in the environment, then that was okay. But if it was just to bring back some rainbow-coloured peacock for someone's entertainment, then it was less okay. Another deal breaker was the absence of clear international or national guidelines on this type of technology. And that kind of debate is playing out a lot at the moment with some of the science coming out of China, not related to endangered species, but this argument that where is the regulation and who kind of has control of that space. And again, if the species totally changed, that would be totally unacceptable. So the second one we'll talk about was changing the properties of natural fibres. So you heard the science angle, or the biophysical science angle from Maddie. Now you'll hear what the public think. So that was what we presented to people. So first of all, we kind of, again, framed the problem. So a lot of textiles these days are derived from petroleum products. But there are natural fibre alternatives, they just don't have the properties that some of the synthetic fibres have. And then we kind of explained the technology pictorially for people. And then we said, okay, if the fibre properties are changed, these are the possible things that you could, get from it. So, some of the benefits. So the number one issue was this reduction in waste. So that, cotton or fibres, natural fibres are biodegradable. And also if they have qualities such as being pre-coloured, then that would reduce the load on the environment in terms of pollution. The technology, they thought it sounded sustainable and scalable. And they really liked this possibility of combining desirable properties into natural fibres that don't already exist. And of course, this is a very relatable product because it affects our daily lives. And it's not something that we're consuming. So it's a much easier pathway to adoption in a lot of ways. But as the survey kind of shows, there's more to it than that as well. Some of the risks that people identified were the possible costs of this new technology. So would it be comparable to the price of cotton at the moment? What would be the potential impacts on farmers who couldn't afford the new technology if it were more expensive or didn't want to participate because of whatever reason? And the fact that natural fibres can be resource intensive, so it's still growing crops and utilising land and water. Some of the absolute deal breakers was if the end product contained allergens. So I don't know, Maddie, does cotton have allergens in it?
Cotton is not allergenic.
Okay. So that was important to people. Unfair accessibility, again, this idea of fairness, so that everybody was able to have access to this new technology. It shouldn't be something that just the rich get access to. And the transference of GM to neighbouring crops. Because it's a lay audience, there's little understanding potentially of how that could happen. The fact that it's a fear speaks to the idea that maybe it's something that needs to be messaged or presented to people so that they understand that process. And the final one I'll present is about reducing mosquito-borne diseases. So this was the scenario set that we presented. The problem is that people get mosquito-borne diseases. There are rarely vaccines for these. And Australia has a region of high risk. So currently there are a lot of manual methods used, but some of them contain insecticides and not really viable at scale. So new synthetic biology technology could enable scientists, as Dan spoke about, to allow mosquitoes not to carry or express the virus. And then we kind of gave them a diagram of how that could kind of scale out. Some of the benefits for the Mozzie technology, fewer deaths and costly illnesses, so there would be less of a health burden. Cross-border benefits to those who couldn't afford the technology. So, there is this, I guess, civic duty that, if a country can afford it, then it's not just benefiting that country, but also countries around the border who perhaps couldn't afford that kind of technology. And environmental benefits, so reducing the use of insecticides, for example. Some of the risks identified for this was this idea of playing with genetics, and if we allow a mosquito not to carry malaria, what else can we not allow it to do? Or how could this technology be used in another species to create something else? So there's a real sense of, like, if you're going to do it, make sure you're doing it for this particular reason, but outside that, it's too risky. Uncharted territory, I think for a lot of people, this is something that's potentially unimaginable, and so they don't really know how to think about these things. And potentially less funding for other solutions. So if you have a mosquito, that doesn't spread a disease, does that mean there's less funding to developing a vaccine for others who might need to use it? So again, it comes down to this equitableness of the technology. Some of the deal breakers, inadequate governance in place, because it is a mobile insect, it might involve multiple countries, and then who has ownership over the regulation or the governance of that. The potential for predators to be negatively affected, and nefarious motivations for the development of the technology. So is it something that is designed to just make money for people, or is it genuinely for public good? And if it is for public good, what's being done to ensure that it is? So there is a real balance between trying to commercialise this type of technology and use it for public good, because I think people don't see them as the same thing. And so it's really important for the scientists developing this to make sure that distinction is as clear as possible. So some of the next steps of our maximising impact work. So as I said, the baseline survey is still in the field, and the analyses will continue on into the new year. It takes a long time to analyse social data. And also, I guess the rest of the first half of the next year will be also a bit of science engagement to feed back some of our results to each of the application domains, incorporating both our qualitative and quant work. Hopefully we can arrange a follow-up web conference with the project leaders that participated in our workshops. It would be nice to touch base and kind of, give some personalised suggestions for some of the things that we found. And ideally have some input into other application domains' impact pathways, because I think that's a really good way to articulate how maximising impact can make a difference into the planning and the development of the science that you do will continue to do. So that's it from me. And Lucy is up next, but I think I have a couple of minutes questions. Yeah.
Thanks, Phage. That's great. One of the interesting trust questions for me is quite often when we develop a new technology as scientists, we try and nuance the wording, right? And I'd be interested in the trust perception of, for instance, when we talk about conservation and genetic diversity, and then said, and what about if that actually meant gene drives, which could annihilate an entire species and might have risks that went on for that spreading? Mm-hmm. How would that affect the trust scenario to the broader conversation, that they feel that they've been, maybe deceived is the wrong word. Do you understand what I'm getting at?
Yeah, absolutely. I think language is really important with this kind of new space. And there's certainly the potential to scare people and use very inflammatory wording. But is that, I guess, the messaging that we want to create or to have a conversation around? So the idea of this initial work is just to kind of float this type of technology and the potential applications it could be used for before we kind of understand how people want to hear more about it. So our survey is also going to ask some of those questions that you mentioned. So we have questions in there asking people, how would you like this technology communicated? Would you like to participate in information sessions about this kind of stuff? Would you like to be involved in decision making? And those who are more engaged are probably the ones who are going to want to seek that kind of information. But I think it's not helpful for anyone, in any field really, to use language that is inflammatory, because that is also deceptive, I think.
Sorry, I might have taken it. I probably said it a bit wrongly. It's not that the language is used to tell fact. It's that more the initial concept may have obscured applications which the broader community would view as too risky or deal breakers, as you would.
Yeah, so you're saying they're not getting the full story?
Yes.
Yeah.
So and in that sense, would that amount to a bigger betrayal of trust than if they were getting the full story straight off?
Yeah, so I think, I kind of roundabout when I answered that question. So I think it is important for some people to know the full story, but not everybody wants to know the full story. And I think the loudest are often the ones who are most engaged. The general public typically wants some level of information and some level of assurance. And they know that it's not their area, and so they entrust scientists to potentially, provide that information for them. But through our work and our constant engagement with people, we'll get a better sense of if they do want to know that full picture, or whether they are happy with knowing that the scientists are happy with it, by proxy. So yes, I get what you're saying, but maybe not everybody wants to know that information.
It's very important that you don't oversell when you communicate. So, because that's what people remember, what you promised. And then when you break a promise, the whole technology is sort of associated with broken promises and lack of trust. And it is important how you express yourself. I mean, you had one statement about gene spreading. So you would talk to them so they could understand how to prevent that process.
Of gene spreading?
Of gene spreading, yeah. As scientists, we don't even know how to prevent that process.
Yeah.
I mean, it's very delicate how you express yourself. And also another question that was with sort of national, international rules. I mean, we don't have those rules, and we're never going to have them, not the next couple of decades at least.
Yeah, of course.
And the same thing with the IPR, it's always behind. So we can generate trust by behaving properly as researchers. I think that's a key way to solve those issues now.
I think that's a really good point. So I should reiterate that these are just lay people's opinions. So, while we know, given that, there is unlikely to be international agreement on a lot of these things, it's the type of thing that people want to happen. So we can take from that information that governance is really important to them, and at least within Australia, we can try as much as possible to achieve that. But you're right.
But I fully agree with your approach. It's just very difficult.
Yeah, absolutely. Yeah.
Spurred by the colossal failure to anticipate public distrust of GMOs, the synthetic biology community has been actively thinking about these issues, at least for the last 15 years that I've been a synthetic biologist. And there was a point when I went to as many meetings about the societal issues related to synthetic biology as I did about the science. And what I see is that the concerns that people have not changed in 15 years. It's still these same things that you list. But what I have not seen yet is a really effective proposal for how to communicate to allay the fears of the public. And so I think that's your real challenge is can you come up with something that the community hasn't found a way to get done yet? Because people still fear what we are able to do.
Yeah, absolutely. And I think it is a number one issue, right? I think the way that we're trying to approach it is softly in a lot of ways, because we're trying to approach it from the angle of where are people sitting with this and how do they actually want to be engaged with? Because a lot of the time we've gone and said, okay, it's obviously about learning more about the technology, that will help. We'll conduct town hall meetings, we'll inform people about these types of things, but they're not effective. And so it's up to us as social scientists to potentially understand how to do that. But I think part of it is also has to rest on biophysical scientists engaging the public early in the development of their technology. So often the technology is at a level and then you take it out and say, what do you think of this? But the ideal scenario is that you co-develop an idea and then kind of work on it collaboratively as much as possible. It's hard to do that, but I think it's the way we need to start working in order to make any type of difference.
And so I guess just touching on that, Aditi, I guess is there a formal, pathway to share the information you're gathering with the respective communication directors that sit within these organisations? Because as papers continue to be published, and I am one of those people, I think it's almost, we sort of almost don't have that control, because Nature's going to put out that media release to go with that amazing piece of science, but we're still gathering the information around how the community feels and how we could be framing those messages. So are we going to work together? I know you and I do.
Yeah, ideally. So I mean, if I'm understanding correctly, so we're trying to, once we analyse information, we want to make it publicly available in some sense. But you're right, like a lot of this informs some of the messaging that our respective organisations could use. And if that's of interest to people, then, we can chat about it maybe after our data is collected. I think last one.
So I think at some point you mentioned that one goal here is to verify people would like to get a bit more involved in what's being done and how to get involved, like how people like, etc. All right, perhaps they don't want to, and that's great. But let's say they do. I don't want to sound crazy, but like, what can we really learn from this that we don't already know, already anticipate? Apparently, we've been doing the same things for 15 years, so they get involved, and what do I get?
So you're asking, what do you get out of engaging with the public?
Yeah.
So, pardon? Yeah, exactly.
It's a great point. If that's the answer, I'm happy with that. But,
I think
not just in the science communication space, but you will be allowed to do your technology in the future and have a career.
Yeah. So the whole point of maximising impact is this understanding that the public, whether you like it or not, has an influence on your technology and whether it's used or accepted in any way. So I think that the value of the FSP is the ability to bring the biophysical scientists with social scientists, so you can ask those questions. But yeah, I think it's fundamental actually moving forward.
Could I just make a quick comment? Having worked closely with social scientists for probably the last, not quite 15 years, but certainly 10 years in the UK, a lot of ground's been covered. The UK and Australia, I know, are different, clearly, but there are cultural similarities as well.
Between a certain proportion of the population.
But I'm just wondering if you guys are just going to go around the same track that we've been around and waste a lot of time and a lot of energy. There's a whole literature out there on social sciences and pedagogy. There are all sorts of really quite serious discussions about the STS guys and girls becoming too embedded in the science and actually, not actually doing what they should be doing, which is, proper social science research, and actually becoming almost like, messaging people for. So there's a whole stuff. I mean, you've probably seen actually So I guess it's coming to Jonathan. What are you going to do unique here, right? What's the difference? I mean, you know, what are you going to do that's not already been done, at least around the world?
Yeah, fair question. I actually don't think there's that much research that's been done in synthetic biology. It's been done in GM and in nanotech and a lot of related disciplines. But
I can give you a load of
that would be awesome. Thank you.
There's a huge amount of work being done, at least by the UK community.
Yeah, so for us, I think what you said is important. So there are cultural similarities, but also a lot of cultural differences in Australia. And when we're talking about a range of different technologies that are potentially suited to Australia, and also the language of communication here is very different. So I think, yeah, it's hard to understand perhaps how we are different, but for us, in the social science community, I think it's very clear that we're trying to develop a process that is collaborative and that engages the biophysical scientists with us. And you say, yes, you know, there are academics or researchers who have embedded themselves in those environments, but at least from the literature we've read, it's not clear what those outputs are. And for us, it's about understanding how people make decisions about these types of risky technology, what drives acceptance, and how can the implementation of these technologies work within the societies that we have here in Australia, and then potentially scale out if necessary. But yeah.
So we do have Sarah Hartley on our international advisory panel.
No, I won't
She'll
go on any more about it.
definitely be
It's
reviewing us
just,
in that space.
yeah, it's there's a lot of people who've got a lot of scars.
Paul and I feel like we have been here. And
I know. So Justine has also done quite a lot of work travelling around to the UK and Europe. And did you get to the States? Not yet. In the responsible innovation space more broadly, and they're all similar themes. There's you get crack on talking. They're all similar themes that they run across in these advanced disruptive technology spaces, and synthetic biology has, I think, some bespoke areas as we have identified. But yeah, we hear you. So please give us that input in the process.
Just add a comment.
Please.
I mean, there's lots. I mean, a decade we added social scientists and humanities as add-on because we were recruited to do it, and there was no proper integration. We have had a synthetic biology center for 10 years now. We have fantastic people from humanities and sociology involved, actually directing our research. We're making products, so we want to make things which will be appreciated when they are there. Politicians are calling either me or some of the social scientists when there's, or journalists, when there's a new GM issue, is this something to take up and put in the news, or is it nonsense? That qualifies the debate an awful lot. So there are many things which have happened and which are different. There are lots of our parties in Denmark, they feel they have handled the GM situation in the wrong way. They actually now like synthetic biology, even though we tell them it's third-generation genetic engineering, just to work with a modular and more precise. We are not saying we don't make mistakes. I think the issues have changed a lot, and as researchers, we have a very, very strong application. But it's true if you just add one and you don't really integrate.
I think the point was in the UK, the STS community are extremely critical of the synthetic biology community, and there's been a huge, what should I say, rupture in some of the discussions and communications around that because they got too close to it, and their community started criticizing them because they were too far embedded into the science, and that's not their job.
No, but it is who gets the voice, who gets the media attention. It's to avoid that those people get the attention.
It's really important. Sorry,
we made vanillin, and we were accused of killing all the vanilla farmers. And then we discussed what is the real issue, and we actually won the discussion. Sorry.
I think these are really deep philosophy of science questions. But just in responding to what we've learned from GM back 10, 15 years ago, I think we know a lot more now, and two challenges remain. How do we integrate science in a way that works? And you just articulated a challenge that's now arising out of an effort to do that. And the other challenge we have is what does engagement mean, and how to engage with not just end users of science, but also with the public. And I think those two challenges remain, and I think we can make a mistake of overcompensating for what mistakes we might have thought with the GM debate and now start going back the other way by providing too much information, for example. But I'd love to have a conversation with you after this session if I can. I'm Lucy, and I'm going to present to you 2 case studies, 2 deeper dives. I'm using a qualitative case study methodology, which uses theory building. And for this piece of work, I need you to suspend the way that you do science as you know it, because this is a very different approach, and I'll explain why that is. So I'm using 2 case studies. It might be a bit frustrating for people involved in either, because I present very little information, but I'm happy to have a chat afterwards. Okay, so just one introductory slide. Unlike the national survey, I'm interested in speaking to immediate end users of our science, which are not the public in most cases. Our immediate end users of science are policymakers, government, industry. Okay, so those, when I say stakeholders, that is the groups of people that I'm referring to. And we really need to understand how they think and their decision-making processes, so that we can get to shape the way that we design our technology, the way we design our science, and the way we design our operations in the FSP generally, but more broadly. Okay, so I think I'm safe in saying that almost everybody in the room has what I call, being a philosopher, a positivist paradigm of science. So that means that you believe that there's a single truth or a single reality, and that reality or truth can be measured. You might probably use a deductive reasoning process to get to that conclusion, and that's one way of thinking about doing social science or biophysical science or bioscience in general. For this particular purpose and for this case study, I'm using what's called an inductive reasoning methodology. The reason I do that is, for this purpose, I have to assume that there is no single truth or single reality, that actually phenomena is socially constructed and continually negotiated, and it can't be measured in the same way. In fact, it simply is interpreted. So that is the approach I'm using for this. That's not the only approach we use in qualitative work or in social science generally. Liz will talk about a very different style of thinking about social science. But for this purpose, I need you to kind of think that this is not a linear process, that this is not a phased process. I am definitely, I can confirm to you, governed by very similar rules of validity and reliability that you might be. And I definitely use propositional logic to get to my conclusions. And I'm happy to talk about my methodology offline. Okay. So the other mistake that you might make, is often made, is that, geez, you've got a small sample size of 8 or a small sample size of 10. You mustn't then be generating a lot of data. But I generate masses of data. And again, we can have a discussion about how I go through that analysis through coding. Okay, so this is the first case study that I'll talk about. And I just wanted to put the interview schedule up there. So these are constructs in which questions for the interview fall from. I definitely want to know how the problem is articulated by whoever I'm speaking with. How do they see the problem? And where do they see their position in that problem? So what's a problem that needs addressing? I also want to get initial reactions to synthetic biology or SynBio applications. I definitely want to understand what the risk relationship is. So often people talk about risk. I want to know risk to whom about what. So really context is very, very important for me. I also asked, amongst the questions, perceived barriers to impact. And I didn't leave that question on. It would be any barriers that came, so it could be technical, social, any barriers that came up, I was ready to receive. And I need to be able to do that in order to build a theory that's testable. Okay, just for information, my informants came from these organisations. I think, this is a preliminary sharing of data, so I could add to this, but I just wanted to show to you that NGOs or non-for-profits were represented in my data set. I spoke to scientists, I spoke to some government representatives as well, and I spoke to individuals. Here are my 5 themes, and I only actually have time to detail 3. So the first one, and they might seem very obvious to you, but I will explain to you how I got to that, where I got. So the first one is protecting biodiversity is the absolute primary lens in which the problem is conceptualised by our stakeholders. So I actually got pulled up when I used phrases like reducing toad numbers, because reducing toad numbers wasn't the problem. At least it wasn't the problem that was conceived in thinking about how others saw the problem. The second theme that I'll talk about is the use of innovative technologies when used appropriately offers hope. So across my entire data sample for this case study, it was an exciting opportunity for all those respondents. And the third one I'd like to mention is that engagement with communities perceived as the key barrier to impact, although the mechanisms differ depending on where you sat. Okay, so the first problem, protecting biodiversity, the first theme to emerge relates to how the problem is framed. So the key theme here is protecting biodiversity is the primary lens in which this problem is conceptualised. And these are the phrases that we used. So protecting habitats, mitigating toad impacts, protecting cultural diversity. And this was not just Aboriginal cultural diversity, this was Australian cultural diversity. Biodiversity was seen as under threat from a number of factors, so land use management changes was mentioned, climate change was mentioned. So it's really seen in a quite a sophisticated manner. So I think it would be a mistake, and I have heard this from scientists working in the cane toad space, to use the phrases reducing toad numbers. That is not how others see the problem. And I've got just a quote there to represent that theme. Australia's wildlife is important culturally and biologically. We have a responsibility as stewards of the environment for others. The second theme I'd like to mention in this case study is initial reaction to SynBio. So I can say this was consistent, that the use of innovative technologies, when used appropriately, offered hope. And really that was founded in an idea that current traditional control methods were not effective at scale. And that was consistent from everybody I spoke to. That was the way that they saw any SynBio, gene editing, novel gene technology. So it definitely offered hope across the board. And some quotes there I've offered, can't see how we're ever going to reclaim habitat from cane toads without thinking about a solution like that, gene editing. Our partners would be 100% open to having the conversation, and I have no doubt that in further development of the technology that safety will be determined or not. The final theme in this case study I'd like to mention is barriers to impact. And this is very interesting from my perspective, also from the MI perspective. Public acceptance and engagement was perceived as their key barrier to impact, no matter who I spoke to. Now, it depended where you sat as to what you thought that barrier was. So if you sat from a perspective of a researcher and sitting in a science organisation, you thought it was very important that you made a distinction, or that whoever was communicating science made a distinction between adding or removing genes, and that was very important to clarify through acceptance. And that again was consistent across the researchers that I spoke with. So here are some quotes. People need to understand shades of GM. Knocking out is not the same as inserting a new gene. The public perhaps are more supportive of selection techniques than slicing and dicing. Over the 10 years, I don't see any change. And just to mention, we can test that in the national survey. We can test whether that is actually how the public think, or what the attitudes around that is. Now, if you sat, if you were represented or a representative from a NGO or a non-for-profit, and if you were really involved at the community level, your main concern or your main thought about barriers to impact was quality of engagement. So how is this going to engage the community that I work with? And often it was couched as in a developing of community activity rather than a communication of science. If you're an NGO that did more advocacy work with the government, you thought the engagement problem was actually about political will and the right policy settings. So here's a quote: "We need to have a conversation and have enough people to participate in that conversation for us to understand the range of views in the community that came from the government representative." So I think there are two implications or two findings or takeaways I can pull out from that I'll mention. The first one is that the cane toad problem is multifaceted, it is contextual and is linked with multiple values. We're not just talking about environmental values here, we're talking about cultural values as well. I think if we present the science as a silver bullet to a problem that is not integrated or not sophisticated, I think we're doing ourselves a disservice, and it's certainly not how others see the problem. And when I say others, I mean those immediate end users of science. And it possibly could lead to suspicion. So I think this has implications for how the FSP communicates the challenge. And I'm sure I just, am speaking exactly the way that Chris thinks, because I think that, you guys have that challenge all the time in terms of trying to communicate exactly to whoever you're communicating what the problem is, what the nuances are, and what the context is. And the other finding, I think, which is more interesting to me at least, is that I often hear the phrase, NGOs are troublemakers. This is from the science community. Well, actually, I don't think that way at all, and there's good evidence to think differently. There's potentially a really unique role for NGOs as conduits between researchers, policymakers, and communities. They are definitely not a homogenous group. They are very highly organised. They have very strategic visions in a lot of the cases. And they can act as potential intermediaries for public engagement. And the one thing that I found that I was absolutely astonished at, because I have only previously spoken with some of the more militant groups like Greenpeace and Friends of the Earth, these groups of NGOs that I spoke with were so keen to be involved in, for example, citizen science or advocacy. So I think that is a take-home that we need to listen to and perhaps stop homogenising this group as being, troublemakers, but also of the same category. So I'm quickly going to move on to the next case study. And this is a very different case study, and I think that's what makes it very interesting. So I've got here that there's a general reticence to participate when I contacted people. What I mean by that is that there was a reservation and a nervousness to speak to me. And I can make some assumptions about what the reasons for those. I would say a few things there. Everyone I spoke to, or at least tried to speak to, very, very, very busy. Of the scientists that I spoke to who were not involved in any SynBio or engineered phage applications, they didn't consider that since I came from the SynBio MI domain that I'd be interested in anything to do with natural phage. And for regulators, it was multiple phone calls and multiple discussions before anyone put their hand up and said, yeah, okay, I think I might be able to speak with you. Because it wasn't a phage is not something that there was an assigned person that I can speak to who knew exactly and was confident enough to speak with me. And I think that also is a lesson for us. This is very, very interesting to me in this case study. There were geopolitical and historical influences on the way that phage and phage science has developed, particularly in Australia, but globally. And that actually impacted on things like how much data was accessible, the quality of that data, whether that data was shared. And even in countries where phage science is much more sophisticated, then there was even, there wasn't a sense that their data was freely shared between countries. So I quickly noticed that to disentangle natural phage science or applications from engineered phage science didn't make any sense at this point. At least it didn't make sense for this particular case study. Oh, here are my phage informants. So you see that there's a difference here, and they're mostly researchers, some regulators, and one private sector partner. Here, there are three key themes to emerge. The first one is, and it is very obvious, but it has implications and also a lot of benefits and a lot of opportunities for us, I think, in the FSP. There's no critical mass of phage research and science infrastructure in Australia. The second thing to come out was that there was a lack of robust data and industry awareness, and that created a barrier to the way that science developed and uptake, and that the tensions were really quite obvious, particularly around IP and funding arrangements. So here that there was no established critical mass of phage scientists in Australia, so phage applications, regulatory pathways and translation prospects are not always clearly defined, despite their obvious potential. And there was no discernible phage science agenda that was actually obvious to me. There's a lot of working around the edges, a lot of working around what regulation might find acceptable, such as compassionate use of phage therapies, for example. And in quite a few cases, the impact pathway wasn't was rather vague and fragmented. The regulatory pathways for sectors external to human health were perceived as easier, although I only actually spoke with one person that didn't do anything to do with human health applications of phage, so that's very interesting. There is as Karen mentioned earlier, a very strong perception in Australia that Australia is lagging behind the rest of the world in phage science. The history of phage development globally has influenced science development here in Australia. Data to strengthen evidence is still emerging. Efforts to create communities of practice are gaining momentum, and this is largely fueled by the AMR crisis. But I think this actually presents us with a huge amount of opportunity for this. So here's some quotes. I'll only just read one. The science is old, over 100 years old. There's a weight of information, even some data, that comes from those countries, mostly anecdotal data. The final theme there I'll just touch on, there were tensions to emerge around IP arrangements and funding expectations. And I'll just read those two quotes because I think they're quite telling. Small startups and small biotech companies will really be the ones to take the technology forward. Big pharma will only invest in that space once small startups are successful in the market. And a quote from the private sector, when you try and have a collaboration, the researcher, sees the company as a provider of the funding. We're not big pharma, our funding comes from public investment. So there's a real tension there to emerge from that. Going to my last slide, because I can see my leader moving. I think the implications for this are very different. The nascency of phage science in Australia provides multiple opportunities, and I really, so this might seem like what I presented as a kind of a negative or a doomsday perspective, but I actually felt there was great energy and great momentum to build communities of practice, and I think that that's what we should ride. In terms of science development, the opportunities of building those communities of practice for science communications, raising awareness among clinicians, professionals and others, a lot of partnership brokering opportunities. I think the challenge directly aligns with the FSP goals. So collaboration, building of capability. This is the case study that really fits nicely into all those things that the FSP vision and goals are built on. the one thing that I wanted to just put here before I stop is that researchers are taking professional and personal risks in pushing boundaries of some of these SynBio applications. So often there isn't a strong network, not often, in this case there wasn't a strong network of scientists, there wasn't a kind of a clear way to think about engagement with publics, for example. So I think we need to recognise that across the FSP perhaps and more widely. And that's it for me. Thank you.
Two questions. Or none.
How about a quick one? So most of that was focused on the health aspect of phage and phage therapy, one would say. But there are other areas, food science, for example, which phage is something that they deal with all the time. And so there's maybe more people, more science, more interaction. Did you actually interview anybody in that realm?
That's a very good question. The case study as it started, I thought and got really excited about looking at phage therapy in aquaculture for food production. And I didn't get very far. It just wasn't, and there weren't enough people working in that space. There wasn't enough confidence to speak about that. And when I mentioned SynBio, people were quite afraid. So, but I did hear, and thank you for raising that, I didn't actually mention that, but I did hear about potential applications of phage outside of health, such as horse racing, domestic veterinary science, aquaculture. So absolutely, but it's just not, that there wasn't a sense that there was enough work or enough confidence in speaking to me about that work. But yes, thank you. I had hopes of actually thinking about phage in context, but it just didn't happen. Yeah, thanks.
I'm just going to interrupt for 2 seconds. We've had a lot of great engagement from the older generation of researchers asking questions, which is wonderful, but I would like to invite the younger scientists to step out of your comfort zones and engage and ask questions. John, thank you for reminding me about that last year. It's in my head, and so please go ahead and ask your questions. There are no stupid questions.
Hi everyone. Wow, really weird experience. Is that sounding okay? Yeah. Or am I too close? So my name is Elizabeth Hobmann, and as Aditi described before, I've sort of been in charge, along with Aditi, in running a national survey, and today I'll be sharing with you some very preliminary results of the survey that's still in the field as we speak. I excerpted an extract on Monday, and it's roughly half the sample that we're aspiring for, so we've got about 4,000 responses. So that sort of means for today, I won't actually present any of the experimental results because the cell size is actually too small in each of the cells, the smallest conditions. But hopefully you will still be interested in some of the descriptors I provide of the bigger sample. Before I start, I do want to recognise that this effort actually, it wasn't just me and Aditi, it was very much a whole sort of FSP effort. There were many FSP scientists who contributed to providing feedback on the slideshows that we embedded within the surveys, and also the survey questions themselves. Also Sarah Combs and the branding team. Where is Chris? Oh, Chris, here you are. Thank you so much. You were just so timely in your feedback and got the ball rolling with branding. We really couldn't have done this survey at all without everyone's contribution, so thank you. So the overarching purpose of the survey was just to provide an overall baseline assessment of what the public actually think about specific synthetic biology technologies. So we went very specific, and in that we presented specific examples of technologies that might be 10 years away from actually being implemented, or 15 years away, but we actually provided specific examples rather than just presenting a definition of synthetic biology and saying, what do you think about it? And that's because people need to have a tangible application to actually form some sort of opinion about it. And the ultimate aim, I guess there's sort of 3 aims there, but for you guys, we're hoping that our results will actually inform your science planning and direction. So really opening that conversation from today, and also when I provide more detailed results next year, we want that ongoing sort of dialogue between ourselves to say, well, what does that actually mean? And we can actually provide you with some sort of insights that might help your science direction. We also included lots of questions about how the public would like to be engaged in the future going forward. So if given this particular technology, would you like to continue to hear about it in the future? How would you like to be engaged? What do you want to know about? So that should inform CSIRO's and the FSP's approach to developing a public engagement process. And ultimately, from our own social science agenda, we want to better understand how people make decisions in complex situations. And synthetic biology is just a perfect example of, it's complex, it's risky, it's uncertain, it's future forward thinking, something that's so unfamiliar to people, it's novel. So how can we unpack what sort of influences people's decision making? And a lot of work has been done in other domains, in risky technologies. So this is sort of like a model that it's called the Technology Acceptance Model from Nicole Hewitts. I'm not sure how to pronounce her last name, but she's at the University of Eindhoven. So I've sort of just grasped this particular model. It melds a lot of the theories together to understand how people make decisions in complex environments and whether they're actually going to accept that sort of risky technology at the end. So basically, I won't go through it in detail, but we're aiming to sort of model this or test this model with our survey data once we have the full set. And I won't be providing you with analysis of that today. But basically, in a nutshell, what it's saying is that humans are complicated creatures. They make decisions in complex situations by, it's not just a rational assessment, they're emotional, they think about what other important people around them are thinking, they're influenced by those people, whether they have a sense of moral obligation to actually accept the technology. So it's always psychological and social factors that actually influence people's decision making. So on the left-hand side, you'll see that there are 2 differently coloured boxes there, and that's where we sought to embed an experiment within the survey itself. And in doing so, we could then tease out whether there are any causal impacts. So we had some hypotheses here in that if we provided people with extra information about the regulation that might surround this technology once it's in the process of being developed and implemented, or not, whether that has an impact on that particular decision-making pathway. So we thought it would lead to greater trust in the regulation and the scientists who are actually doing the research. Then we had a second experiment where we again provided information about, sorry, I'm running out of breath. We provided extra information to survey respondents about the potential for the public to be engaged in the future. So would they like to participate in future research, that there would be that opportunity. And then we had another condition where we didn't provide that information. So we could test it just like an experiment that you all do. This is just a sort of a diagram of our research design. So Aditi described to you earlier, and she presented you a few of the slideshows. So we actually had 7 different slideshows to represent each of the different synthetic biology technologies. And so we have a big sample of 8,000 who are being randomly assigned just to receive one particular synthetic biology technology survey. Okay, did you want me to read out those topics, or you yeah? Okay. Then further beneath that, within each of the survey topics, we had 4 different sort of conditions, and they represent the amount of information or the type of information that we presented on each of those topics. The control is pretty much just presenting them with information about the problem, current methods for addressing that problem, then introducing the synthetic biology technology, and that's it. With the regulation, it was that extra slide on regulation. Engagement was the extra slide on engagement. And then we had a full factorial, so we had both added at the end in the 4th condition there. Okay, so preliminary results. So we currently have almost 6,000 responses, but I'm only presenting you with the results of a smaller sample today. This is very consistent, so this is knowledge. So before we even presented anyone with a definition of synthetic biology or the slideshows, we asked people how would they describe their knowledge of synthetic biology. And this is consistent with research overseas as well. As you can see, the vast majority have either no knowledge or just a little knowledge. So there's nothing really new there. I have drawn out an implication there, but obviously there exists an opportunity to communicate with the public, to raise their awareness. So that's sort of quite obvious. Oh, I should have done like a drum roll. So this was one of the variables we included in the survey, and I know that you would all be really interested in this particular variable, but for us as social scientists, there's a lot more in the survey, but I thought I'd present you with what you are most interested in, but then also tease this apart a bit by showing you some data on other questions that we asked. So you can see here, we asked people after they saw the slideshow, and remember that the slideshow, it's quite a visual slideshow, presents the problem, the solution, doesn't go into a lot about the risks or benefits, or it doesn't go into any of the risks, so keep that in mind when you're looking at those results. They are more positive, so there's more people who are supporting each of those technologies than there are who are not supportive, on average. For each of the technologies, I can do a deeper dive, and you can have a look at, obviously, the frequency distribution, but on average, that gives you an idea that generally people are supportive based on receiving that minimal information in the slideshow. Okay, so one thing that sort of struck me when I looked at that was that, there seemed to be a general trend that if I reorder it in terms of support, and again this is consistent with research overseas, that generally people are more supportive of technologies that don't involve a target of a large animal, particularly cuddly one maybe, like a chicken, or an invertebrate. They don't really care about mosquitoes, maybe. So that was an interesting observation. But then when we looked at other data on a few of the technologies in here, we realised that we couldn't really make that sort of global observation or assumption. So for example, with invasive pest control, so you can see that support is 3.6.
What are the units?
This is a scale. So it's a rating scale.
So 1 is would not support and 5 is would strongly support.
Okay.
Yep, sorry. Yep, on the y-axis. Okay, but with invasive pest control, we actually followed up, so only with this particular survey. We asked a lot of follow-up questions, so there's more in the data set, but this is sort of a critical one to show that actually support obviously is quite variable depending upon the target pest. So, yeah, no surprises there. Cane toads, support's quite high, but for things like kangaroos and wallabies, possums, maybe not. So the implication of this for us as we go forward is, you can't just provide a general positive sort of explanation of the technology and then ask people what they think and go, oh, they support it. You need to have more of an in-depth discussion. So it's more about, well, where is it going to be used, with whom, how is it going to be done? So we need to have more of a deliberation with people, and that's difficult to achieve in a survey, but maybe going forth, CSIRO can invest more in those sorts of methods. Another example, so back to our sort of ordered graph. So looking at one on the other end of the spectrum with reducing pollution by creating pseudo-organisms to sort of not metabolize, but to break apart chemicals in the water is quite strong overall. However, we also made it real for people. So we said, to what extent would you be actually willing to swim in that waterway where that pseudo-organism had been used, or eat the seafood, or even drink the water? So we made it really sort of salient and personally meaningful to the person. Sorry, stop it. So you can see there, that was really dropping off compared to what it was before, well above the midpoint. So the implication, again, similar to what I said before, it's really important to engage people at a more local, personally meaningful level. Okay, so this speaks to, you know, where you are wanting to introduce the technology in the field. You need to do a lot of community consultation before even considering introducing it. And it may well be that, you know, it might actually end up that pseudo-organisms are introduced, but maybe in waterways where, you know, people are not around, and so it's purely for biodiversity and conservation purposes. Oh, another sort of interesting example along the same lines. So again, in our technology slideshows, we didn't go into the specifics of the genetic techniques, but later in the survey we asked people, just thinking in general about synthetic biology technology, to what extent would you support research that, you know, does these sorts of things, changes an existing gene within the organism, removes a gene within that organism, or adds a gene from the same species or from a different species, so transgenics. I think that's really. So yeah, support sort of dropping off quite a bit there compared to the support ratings for the technologies. So, you know, once you actually articulate to people what it actually means on that genetic level, it's a different story. So I just made a provocative implication there. I'm not saying that that is what you should do, but it's one of the options. It could just mean, look, we need to actually explain in greater depth what these genetic techniques are, or we could even decide, well, is it important for people to know? Maybe it's more important that they just know about results from trials in the field. They've seen that, you know, the risks are not there. There's lots of benefits. That's all they really want to know. We don't. It's still an open conversation.
Are these differences statistically?
Yeah, good question. I did do some statistical testing. With this one here, it was tricky. I did try to run it. Sorry if you're not really a stats person. So I did I had to manipulate the data, and then it was iterating for so long. So I turned it from wide to long to do a repeated ANOVA. So it actually didn't work. I did individual t-tests instead, which isn't great, and they were all statistically significant. But that's by virtue of the large sample size. For me, if I look at the difference with the last one, to me that's the most sort of practically significant result.
If they don't know much about SynBio, how much do they know about genes? If you have a question like that,
yeah, exactly.
How would you rate that? How could you
Yeah, fair enough. I think that's a really good question, and going. Oh, yeah. Sorry.
Sorry, this is so interesting.
I know, there's so many questions that a survey, you can't address everything in a survey, and it's so frustrating. It raises more questions than it answers. Oh, okay, so the last, how am I going for time? Okay, so I've got 3 more slides, and these ones speak to that other purpose we had of, you know, how to design a public engagement sort of process. So we asked people near the end of the survey, what's your belief in the appropriate level of public involvement or in making decisions on this particular technology? Can see that the majority are sort of in the middle there. They just choose one option, sorry. So we have 77% who just want to be kept informed or consulted with and opinions considered. Small proportion, but still, 12% is a fair amount, wanted to be directly involved in making decisions. Now I would anticipate, so remember that this is a national survey. We've given people sort of very new technologies, unfamiliar concepts to sort of comprehend. It's very distant in their mind. Whereas if we actually did this survey, say, on a local community who was going to be affected by some sort of technology, I think we'd be seeing direct involvement shoot right up. Okay, so you need to think about these survey results in the context in which, well, in terms of the sample and the context in which it was delivered. We also asked people about how they would like to be personally involved in the future, and again, we're not seeing an overwhelming desire for lots of engagement. Seems to be more of a lighter touch at best in terms of just engaging through social media or receiving a summary on the results. But overall, I think what we can see from these results, and I'll need to unpack it a lot more, is that when we engage with the public, it's not a one-size-fits-all. We need to have a multifaceted approach with different segments of the community. And I'll finish on a positive note. I do have a cotton slide as well, so I can bring that one up as well. In terms of trust, so we are, so the blue line is just sectioning it off because the first two options were with regard to the technology. So we asked people, to what extent would you trust scientists working on the technology, that they would do so responsibly? And also, to what extent would you trust the government agency responsible for approving or regulating the technology? So on the left, reasonably supportive. There's more people who, not supportive, trust. There's more people who trust the scientists than not. But government agency is falling off a little bit to just at the midpoint. And then we just said in general, how much do you trust scientists and university scientists? So I think from this we can see that the public do have general confidence in what we're doing. And next up is Alison. I can show you the slide today. Yeah, so your question, I think, as I said during the presentation, you've got to interpret the results in the context. So we didn't provide any information to people about the genetic techniques at all. So that's really like baseline or zero understanding of what it actually involves. And I would really love to do a study where we actually provided a bit more information, maybe in a visual sort of format, to see how people would respond in that instance. Yeah.
The trust one's very common.
Is it?
Very similar to what
yeah.
For some reason, the public really trust scientists. Quite interesting what happened yesterday as well. I wonder whether that trust will be eroded 2 days ago.
Yeah. Questions? Who's managing questions?
I assume a lot of these similar surveys have been done on GM technology 10, 15 years ago, or maybe stem cell research. At some point, are you going to overlay your results with these previous surveys and then see how SynBio is maybe similar or different in specific aspects?
Well, that's a really great idea. We've been so busy in just getting the survey in the field, I haven't really even thought about what's the next steps, but I think that's something that, should be done, just to see how it compares. Although, different contexts, so obviously we try to find results in the Australian context.
Do you have any data on the participants? Because I wonder if there's variables like age that might suggest how bright the future is.
We do have some demographic questions in there: the standard age, gender, education. What else do we have? Oh, income, household income as well. So we can look at that in greater detail and even do some segmentation.
I think that would be really interesting.
Is there any way of knowing how much people lie in this? How much they say that they're
Yeah, that's a
clear about
fantastic
something
question.
that they really don't, or what they say, I will not drink water from here, but then they will?
No, that's a good question. Look, apart from putting a lie detector scale in, it's really difficult to sort of, pick up on that. We did attempt to include some questions on comprehension, so that would sort of measure how much they actually understood the information in the slideshow. And then at the very end, we did ask, to what extent, how strongly do you feel about these issues? And I think generally people would probably answer accurately to those questions, and then we can see if, people who didn't feel strongly, they don't care, maybe they responded in a totally different fashion to those who do care strongly. So we can do, comparisons between different types of people. But yeah, we didn't have any sort of lie detector scales in there.
And it is human data, so, we're all...
Yeah, but maybe there can be a control question that you
Yeah.
know what the answer is.
Look, I think, and like people do have that social desirability bias, but I think in an online survey where it's anonymous, that's sort of lessened compared to if say, someone's doing a survey in front of me as the researcher, I'd expect quite a lot of social desirability happening there. But in an online anonymous environment, people are generally going to say what they think because it's anonymous.
Probably one more question.
Yeah, I was wondering if you had considered perhaps including a objectively terrible synthetic biology idea in your question to act as a negative control, to account for things like perhaps your own unintentional persuasiveness with the slideshows, or even I've noticed that a lot of the values are hanging around 3, right, which could be maybe there's a general level of acceptance, or maybe there's a level of perhaps social apathy or confusion, right? So if you included a question that was like, this is a terrible idea, right, and you still would see values hanging around 3 that might suggest maybe...
Where were you when we were designing the survey? we'll take that idea on board. And it's very creative. Yeah.
So that was the grand mean, so that's probably why it's kind of sitting in the middle, because we haven't teased out the experimental design part of it.
Oh, yeah, so we do have the means for each of the technologies, but I don't have the means, I didn't show the means for the individual information conditions. Yeah. Something for us to think about next time. We do another survey. Okay. Yeah, feel free to come up to me later tonight if any more questions.
Because what I found when they
Is that about right, volume-wise? It does feel very weird. I had to do some psych testing when I went for a job in the Commonwealth Government, and there are totally questions in there that I think were designed to see if you'll lie. Like, I never hit anyone when I was a child. And I'm like, no, I never did. I never hit my little brother or vice versa. Anyway, I wondered later what they would have learned from that test. Okay, so this is an overview. There are other good ones, I'll tell you more about it later, but I would love to know what the psychologists learned about my psyche. Anyway, so I'm a legal researcher who also has a background in cell and molecular biology, and what I like to do is play at the intersection of those two things. So what I'm going to talk about is two different approaches to IP in synthetic biology that we can observe, and why that's significant for the future trajectory of the field. I'll talk about some potential patent problems that we can see being theorised about in the literature,
And then I'll turn to focus more on the Australian context, because there really is no literature and discussion out there, at least in the academic legal research, about IP in synthetic biology in the Australian legal system. And I'll talk about a new project that I'm just starting with a project grant from the FSP. Thanks, by the way. Okay, so these are the two diverging approaches that I was talking about on either side. So everything else that's in the public domain is in the middle. So on the left-hand side, you can see we've got the approach where IP rights are sought in synthetic biology, and they are seen as really important and crucial to promote innovation in the field and to get the useful, beneficial products that we want to see. And in that approach, things like synthetic genomic technologies are seen as very much rightfully the subject of patents, because they're seen as a result of human intervention. They're not things that are just naturally occurring, they're the result of human invention to transform natural things into something different. So they're seen as appropriate subject matter for patents, and patents are seen as really important. And there's a couple of examples of things that are patented. I learned earlier in the week that some GFPs are patented, including one that a scientist got out of an online registry that he thought was all open and free to access, but then got asked for $100,000 by a patent holder. CRISPR is patented. Until fairly recently, the breast cancer gene sequences were patented. And then the commons on the other side is it's a similar shape and colour to the public domain, but it's a bit different. So in the commons, there's no private ownership, there's no IP, or there's a decision not to exercise IP rights. But it's not a total free-for-all. So there is some control, usually a collective group set of rules, basically, to manage the resource, the data, and so on. So open source software development, Creative Commons are examples of that. The Open MTA that Jim and colleagues have developed, that Open Plant and the BioBricks Foundation have developed, is a tool to sort of help to create a commons and make sure that those tools are accessible. Whereas in the public domain, there might have been rights at some point, but they're now expired or they're waived. So we don't have to get anyone's permission to access things like the works of Beethoven. They're just out there. There's no copyright and so on. So public domain and commons are a little bit different. And the commons have specific goals. IP theory, it's a different beast from the public domain. So why should we care about any of that there are these two approaches? So science and technology studies experts, Jane Calvert and Emma Froh, pointed out that these are really two contradictory understandings of the relationship between IP and innovation for beneficial use. And what's interesting to me is that tension between those two approaches has been there right from the beginning of the field. So even in the sort of early to mid-noughties, when people were starting to talk about synthetic biology, the idea that some scientists wanted to share and make things open was already present. And it really came from concerns about patents possibly getting in the way, or restrictive licenses getting in the way. So what I'm interested in asking is, can both approaches be right? Can they coexist? They kind of do at the moment, in what Jane called a diverse ecology. But I'm interested to know more about what it means that there are these two approaches. So one of the reasons that we have regulation and law around emerging technologies is to support desirable innovation and benefit sharing. The other reason we have law and policy is to assess and manage risk. And that's what we tend to focus on when we think about law and science. But I actually think thinking about how to promote the benefits and promote innovation is more interesting. The legal system does that by having an IP system, having patents. Ideally, patents are meant to promote innovation and the dissemination of useful things for the social benefit. But legal tools can have
off-target effects, just like biological tools can. So what I want to know is the IP system having some unintended consequences? Is that why scientists are concerned about patents getting in the way? Does that mean that we need to change or fix something in how the patent system works or how we go about using it? So we have lots of theoretical problems. In the literature, these are some of the problems that people are thinking might occur in synthetic biology. Many of these problems were debated in relation to human gene patents, embryonic stem cell patents, and other things. So potentially we could get a problem that I've called microbe-soft. I borrowed that word from ETC group. That's where you get a patent really too early in the development of something, so it's more like a patent on planned research, and it's so broad and so foundational that anyone that wants to work in that field needs access to things that are within that patent. That's potentially a problem. The anti-commons is sort of like the opposite problem, where you get lots of narrow patents. So potentially if you're building a complex technology and there are patents on many different components or parts, that could cause a problem because you have to find a way to negotiate access to all those patents. And that's something that we've seen happen in the IT context. The third one that we see in the literature is the possible problem of standards, of creating a patent ambush. So we know standardization is really important in synthetic biology, and this theoretical problem would occur, and this also comes from IT, where if the community were to set a specific standard for doing something like assembling biobricks, and then it turns out that some bit of that is actually patented, the patent holder can then basically hold the community to ransom if everyone's already using the standard. It'll become a problem. So, who's an IP specialist, pointed out there's really two issues here. What do we grant patents on? We have to make sure patents are high quality, so they really meet all the criteria of inventiveness and novelty and so on. The other issue is, what do patent holders choose to do with them? So even if someone had a very broad patent, but they chose to license it freely and not for an exorbitant cost, it might not become an issue. So there's really two sides, two different levels at which we could look at the problem. We also have theories that suggest that if people share their work in a commons, we should see more innovation more quickly. I did have the chance to talk to Jeff Booker earlier in the week, and he said that he thinks if they had not put the yeast, synthetic yeast data into a commons and shared it, they would not have made as much progress in the last decade as they have, so that was really interesting. These theories come from other technologies, so von Hippel's theory about democratizing innovation mainly comes from software development, also sports equipment. Who knew there's user-generated solutions in surfboard design innovation communities as well? He found that when some users are willing to reveal what they've come up with, everyone else can improve it, build on it, debug it, and so revealing it actually can increase the value of the information. And Ostrom's work draws on another type of resource. So she was looking at what happens when you have an environmental resource like a fishery area or a water catchment, and the users of that resource manage it in a group or collective way. They have some sort of organising rules. She found that when that works well, the joint return is higher than if everyone just did their own thing and they didn't share control and follow the rules that they agreed on amongst the users. So it's a different type of resource from IP, but there's actually a lot that we can learn from that theory, I think. How am I going for time? We're running out. Okay. So as I said, there's really not that much has been written, at least in the academic literature, about how this is actually playing out in Australia. There's lots of research about IP in synthetic biology in the States, and I've spent some time looking into patent applications and case law in the States. But now I want to turn and look more at the Australian context.
And the Ecola report had some findings on IP. They found that at this stage the pattern of patent activity is similar to the US, but it's a much smaller scale, as we might expect. And the patent systems, the legal rules around patenting are actually quite similar between the US and Australia. In the US, we do see lots of patents being granted on processes, like methods for constructing large assemblies of DNA or constructing the synthetic cell. Methods of making isoprenoids, things like that are patented. So we can expect that kind of thing to start happening here. They did point out the particular issue with standardization in relation to synthetic biology. It makes it a bit different maybe from some earlier biotech discussions that we've had about IP. So they suggested it's really important for us to engage with standard setting organizations. They noted that in the community we've been talking about standardization and open sharing and also IP. And they also mentioned that to be competitive we need to strengthen our culture of technology development and commercialization, and a well-protected IP base is part of that. They also raised some other interesting questions that go more to the issues of access and equity that some of the other speakers spoke about. So they asked a question about whether IP could end up meaning that ownership of synthetic biology technologies is concentrated in a few companies that are all in the global north, and will the benefits flow to other places? So that's a really important question as well. This is some more context in Australia. We do have this experimental use exemption. So this means that if you are doing experiments on the subject matter of a patented thing, you're not infringing the patent. But it's not as good as it sounds. I put some examples there. So you're allowed to tinker with the patented thing to see how it works, see if you can make it better, to work out maybe if the patent is even valid. But that doesn't include using it as a research tool in other experiments. So, we know, because this has played out already in the States with the very same phrase in the law, this doesn't apply to the use of patented research tools. So the experimental use exemption that we have, it could fall short of providing researchers with appropriate levels of protection from patent enforcement action. And so that is an important matter that remains to be addressed. So this is from work of IP experts Jane Nielsen and Diane Nicol and colleagues. So if we're going to address that, we need to know what is actually happening in terms of synthetic biology IP. We've got the theoretical putative problems that I talked about, but we need to know what issues are scientists actually having. So in my project with the FSP, I want to do a few different things. One of them is to look at different sharing initiatives across biology and maybe even other fields of science. See if I can identify common features of successful initiatives and to determine what are the criteria for success anyway. But I also want to do empirical work with scientists and legal managers to understand what's actually happening. The theories are all very nice, economic theories, but we know that doesn't necessarily translate to practice. So I want to know what experiences are scientists having with the IP system, what is their approach to sharing, what do they need in terms of infrastructure, and another step will be to do patent landscaping work to analyse what patent applications are actually out there in Australia and what's being granted. So the qualitative part of this will be interviews with scientists and legal managers. I've planned to do up to 25 interviews for about 1 hour. They'll be semi-structured with sort of open-ended questions. Initially, my idea was to start with the foundation technologies domain, but I think it sounds like many people are doing work that uses components, basic biological toolboxes, even if you are not specifically sitting in that domain. So I think it can be broader than that. So I want to talk to people both in CSIRO and also in universities and commercial collaborators. And in using that interview data, I will ask very targeted questions to IP Australia's Patent Analytics Hub, because patent searching is a horrible thing and nobody should ever do it. So I will outsource it to those people. It's a hugely technical time sink. But I will use what I hear from you in the interviews to work out the right questions to ask, to interrogate the patent analytics. And then, using all that data, I can come up with a survey with Aditi's help and Lucy's help also, hopefully with the qualitative part. And I'll seek a broader range of responses with the survey. So the goal of doing that is basically to investigate what's going on and to make recommendations to see if there are legal or policy changes that would help. To make recommendations about the structure of sharing mechanisms. Maybe develop some sort of practical tools or handbooks that are useful for scientists in the field. Of course, I have to write journal articles, so that's up there as well. And what I think is really important is to do it in an interdisciplinary way so that we have inclusive discussion between disciplines. Otherwise, it's just going to be legal academics theorising about what might go wrong and theorising about what might help. But if I can actually talk to scientists, we might be able to get some better practical data and solutions. So my goal is to help basically design the regulatory environment so it does have that effect of supporting development of the industry and maximising the beneficial innovation that we get out of it. So I encourage you to join me. I'll probably write to you very soon, or not till at least midway through next year. But even if you don't want to do a formal interview, I'd love if you want to chat to me informally about your experiences with IP. That would also be wonderful. So, thank you. Oh, I got a thank you slide. Yeah, I wanted to thank the FSP for the grant and Claudia for leadership and guidance in this area. And Aditi is my CSIRO mentor. And I've also got a UC mentor, David. And I want to thank Barry Pogson for teaching me genomics in 2002. It's a long time ago now, but I remember it was a fun class, so thank you.
So as we identify projects with actual protectable IP that we think that is protectable, can we send these people to talk to you to work together? That would be well.
I mean, I'm not a patent attorney.
Aware, yeah.
Yeah, but yeah, I'm certainly happy to talk through the sorts of issues, the questions that you might have.
Yeah, we have patent attorneys.
Yeah.
Yeah. That would be great.
Are you going to be making recommendations to the platform as to how they might best structure themselves, particularly in terms of the high-level aspiration that we're entering into alliance-style relationships with big organisations and how we manage IP ownership and sharing, and also how we deliver against the platform's goal to make foundational capabilities widely available and not wrap up IP ownership in one partner or another? Long question.
It's a really difficult question because we do have, like I said, that tension between expectations of. It says it's going to shut down in 5 minutes. Yeah, so there's expectations of sharing and community sort of endeavour, but then we've also got commercial partners, and they might have their own expectations, or even other funders might have expectations of patenting. So I'm not sure I have an answer to that right now, but it's certainly something I want to think about. This is a 3-year project, so yeah, thinking about how to navigate that is a really important question, I think. There was another part of your question and I've forgotten.
I think I forgot.
That's right. We'll talk later.
Just thinking, one of the things that seems to be quite important is this idea of developing commons to promote entrepreneurship and innovation with people who don't have large resources and want some kind of guarantee that they can get investment without being frozen out by conflicting patent claims, etc. And so the issue of establishing some sort of triage and exchanging nuts and bolts components can be problematic, as you just said. But one thing which is a clear avenue for exploitation, which is not really well explored yet, is looking at expired patents and looking at materials that are off patent and accessible. And there's been lots of discussion about trying to make some kind of database or file that would allow access to those. At the moment, it's very difficult. You individually need to go into the patent literature, look at expiry dates. It's not really accessible that easily. So something that the FSP could actually contribute, even locally, would be some kind of portfolio of materials that would link into what people are using or want to use and make that accessible.
Yeah, it's definitely one of the biggest problems is figuring out what's patented and what isn't. So if you get something from the Firebricks Foundation or the iGEM registry, it doesn't say on it that it's not patented. So you would have to check yourself.
But you theoretically just do it once, and then there would be a massive knock-on effect.
But I hadn't thought about looking at what's off patent. I think that's a really interesting idea. I mean, that's how we come to have more affordable drugs because they're off patent now. So it's an interesting thing to think about.
Last question.
I have something I don't understand is for the patent, for example, the CRISPR patent in US. I don't know whether the CRISPR patent has been raised in Australia. So apparently a lot of people is using CRISPR. So how this patent can be protected in Australia?
So is Perry So Perry has been looking at getting licenses to use CRISPR and figuring out what the story is around the CRISPR patent and how that's going to tie into CSIRO's work. So she's probably a better placed to answer. But if they are geographic things, so you can have a US patent, an Australian patent, they're different beasts. So if you wanted to enforce your patent in both jurisdictions, you'd have to apply and get granted a patent through both legal systems.
So we do, as you know, have someone who's done work packages investigating SIRO's freedom of operating in the space. And I was going to ask her to just write a one-page brief for us, so we can probably take that away.
Thanks, Alison.
Okay, so we're half an hour behind schedule. Next time I've got to schedule more time for you guys.
I think that's the late coming in.
I didn't know. It'll be late for me. So definitely more discussion time for the social scientists next year. Please remind me when we get to the planning stage. Now we have poster session, which is one of the most important sessions of any conference. We set up across the road. Those of you who've been over there already, can you please be the Pied Pipers for everybody else to follow? We have a dinner setting at 7.